for the 2021 Annual Science Symposium that we host each year in the Center for Research on Ingredient Safety. And what I'd like to do, because I think probably a lot of people who are joining us virtually are not familiar with our center, let me just take 1 minute to tell you a little bit about it. And so this is a partnership between academia, industry, government, and non-government organizations. And we're primarily focused on chemical-based ingredients and safety. And we purposely made this a multi-sector center, trying to involve really the key sectors that are involved in determining health issues around chemical ingredients. And primarily what CRIS is, it was established to serve as a hub for objective science that adds rigor and data to highly visible discourse on consumer products and ingredient safety. And as we all know, this happens a lot, primarily through a lot of information and misinformation through social media. And so we believe that an academic setting to bring the different people from various sectors together to discuss some of this discourse is really the place to do that. And so the mission, the CRIS mission, is to conduct research and to provide insight on the safety of ingredients in food and consumer products to support, and I want to emphasize here, evidence-informed decisions by consumers, industry, and policymakers. And so, as I mentioned already, we do host an annual science symposium each year. And this year's symposium is focused primarily on ingredients and packaging, the safety implications, and so forth. And so what I wanted to do is just kind of put some of this in perspective today. And I'll just use myself as an example. When I go to the grocery store, for example, I'm not really concerned about, in fact, I don't even think about the packaging. I'm really interested in what's inside those packages. And if we stop and think about it for a minute, you know, packaging is really important. Not only does it define the amount of whatever it is that we're buying, it certainly keeps, especially if we think about food and beverages, it maintains this, maintains that they're sanitary. And also it maintains the integrity of the product. You know, we just assume that when we buy things in the store, they're going to be okay. And there have been a lot of advances in technology which has extended the shelf life of many of the products that we buy. However, what I think don't think about very much is that those food products come in contact with that packaging. And that raises a lot of questions, things about, well, which specific chemicals are in that packaging? Also, are those chemicals actually migrating into the food that we're purchasing and using at home? And if it is migrating, how much of that is migrating? And is it safe? And so those are some of the questions I think we're going to address today. The other part to this is that recently there have been major technological advances in packaging. And a lot of this has been possible due to the unique physical and chemical properties of nanomaterials, which are now widely used in packaging. And these nanomaterials have unique properties, but there's also a lot of questions about their safety. And so today's first session in our symposium is really going to focus on nanomaterials in these packaging products. And, you know, the other thing that we probably should be thinking about is a lot of the food that we buy come in packaging that is basically ready to use. We often warm our food or bake our food in that packaging. I mean, that's for consumers' convenience. But again, that adds another dimension to the things that we're purchasing and using. And so the first session today will primarily focus on nanomaterials in packaging. We'll talk a little bit about how these are regulated and talk about it from a regulatory perspective. We'll talk a little bit about the technology around it. Do these materials migrate into food? And then in the afternoon, we will focus more on contaminants in packaging. Also, those contaminants which are added in a non-intentional way. They're part of the materials that often we use to make packaging, but they're not 100% pure. There are other things in there. And then we'll finish up by talking about sustainability. I think about when I wheel my garbage out every Tuesday morning and that garbage can is full, probably 90% of what's in there is packaging. Anybody that buys anything from Amazon, you know that you buy something that's small and it comes in a huge box, and that's happening throughout. Even when we're buying groceries, it results in a tremendous amount of waste. And so we'll have one speaker talk a little bit about sustainability. And then the last thing I just wanted to mention is that a very important part of CRIS is communication, risk communication to the general public. And so each year we make a point to feature at least one speaker that will talk about science communication to the public. And we will again do so this year. And so with that very brief introduction, what I'd like to do is introduce our first speaker. Our first speaker is Sabina Halappanavar, and she is a research scientist with Health Canada and is also an adjunct faculty member in the Department of Biology at the University of Ottawa in Canada. And so with that very brief introduction, if we get Sabina's presentation pulled up, which I see we have, we'll turn this over to Sabina. Thank you very much, Sabina.
Okay. So good morning, everyone, and thank you, organizers, for the kind invitation to present at this meeting. I was supposed to be there in person, and I apologize for not being there, and I hope that in the near future I'll be able to visit you in person. And it was really nice to see so many of you without masks roaming around and giving me hopes that normalcy is coming back soon. So yeah, my task this morning here is to initiate some discussion around the challenges we have experienced assessing nanomaterials safety and point out to the opportunities there may be in continuing to assess them. I just wanted to let you know that I'm not a food toxicologist, and my presentation is not going to touch at all on the packaging issues, but it's going to give you a brief overview of the general issues and challenges and the opportunities that there may be and can be applied to all sorts of fields. So what are nanomaterials? By definition, Health Canada considers if a substance is at or within the nanoscale, which is defined as 1 to 100 nanometer, in at least one external dimension or exhibits one or more size-associated properties, by which I mean the properties such as melting point, electrical conductivity, optical or thermal, magnetic properties which change in the nanoscale range, then the material is considered as a nanomaterial. And European Commission also uses the same definition for nanoscale, which is 1 to 100 nanometer, but their definition emphasizes on number size distribution. And if substance has in an unbound state or aggregate or agglomerate state 50% or more of the particles in the nano size scale, then a product or a substance is referred to as nanomaterials. So in general, I mean, as such, we are not really new to nanomaterials. we do harbor some of the nano entities within our body. We are exposed to naturally occurring nanomaterials in the form of pathogens and other examples here. We are also exposed to, on a daily basis, to nanoparticles or nanomaterials generated as byproducts of the processes, incidental processes such as combustion, degradation of paints and pigments, etc. But then we have added the third category here, which is the new category of nanomaterials, which are called manufactured or engineered nanomaterials. And they are made to be in the nanoscale purposefully to harness the size-associated properties. And these come in shapes, different shapes. On the top right corner is the graphene sheet, and on the bottom left is the zinc wire ball. And the image next to this ball shows the network of these nanowires in the ball, which is very, very interesting. So in addition to the sizes and shapes, they also exhibit different crystal phases, surface chemistry, and their surface can also be functionalized by adding different chemical groups. So this variety in shapes, sizes, and tunability, and the possibilities of combinations of variations of these properties makes the nano universe gigantic and a kind of nightmarish scenario for toxicologists. And they are readily incorporated in wide variety of things, as the organizer was just speaking about. And that has actually increased our exposure to these materials in our environment, and thus the concern for human and environmental health is also rising. So then what's really the concern? The concern is very, very simple. In simplified manner, we can explain it as when you inhale a smaller particle, for example, you have here human lung and different sections noted, and the smaller particles are able to evade the organism surveillance and infiltrate deeper into the tissues, the areas of tissues that are not accessible to larger particles. So as you see in the figure, small particles get down to the alveolar region where clearance mechanisms may not be really effective. So the smaller size also increases surface molecules. And so increased surface area results in larger chemical and biological reactivity. And this could be negative or could be positive. In the bottom graph, you can see that the left hand graph shows you that in the nanoform, titanium dioxide nanoparticle is able to induce the increased amount of inflammation compared to the bulk titanium dioxide particle when you express the dose in the mass units. But of course, when you convert that mass to surface area, deposited surface area, it seems like the observed effects or the difference in the effects between nano and bulk disappear. And so dosimetric is also important. And this was all back in 2005, we already knew based on the particle toxicology. So when particle toxicologists started working on these so-called new engineered nanomaterials, they already had a lot of scientific background on what to expect from these materials. However, because they were newly synthesized in the new nanoscale range, even though we had accepted that the traditional human health risk assessment of nanomaterials was very well applicable to nanomaterials risk assessment, but the data requirements were still there, that we still had to address some of the basic questions as whether or not these nanomaterials toxic, what type of toxicity they do induce, is it different from the bulk materials, and the other questions about the 5 pillars of the human health risk assessment had to be answered. So to do so, the OECD initiated a sponsorship program in 2006, and the main objectives of which were to establish an OECD database on manufactured nanomaterials, and also test some specific nanomaterials for their human health and safety evaluation. And so this program targeted specifically 13 of them. Initially there were 14, but then they were reduced to 13 of them, which are shown here at the bottom of the slide. And the one of the other important objective was that to evaluate the appropriateness of the existing test guidelines for chemicals for their applicability to assessing nanosafety. And 10 years later, in 2016, a paper summarized the major outcomes of the sponsorship program. You can take a look at this paper, which has many, many details of the endpoints, assays, methods that are tested as part of this sponsorship program. But the main conclusions were that the approaches for the testing and assessment of traditional chemicals were appropriate for assessing the safety of nanomaterials, but the data suggested that they have to be adapted to the specificities of nanomaterials. And this is very important. The specificities of nanomaterials is something that we still don't know. Therefore, how to adapt these test guidelines to nanomaterials is still a work in progress. And it was also suggested that some endpoints of relevance for nanomaterials differ from those typically considered for traditional chemicals. So meaning new test guidelines may have to be put together for some of these new effects. And lastly, the most important highlight of this work was that after 10 years of work, the data still suggested that there were no standardized methods or protocols available for even sample preparation and dosimetry. They still needed to be developed. They still needed to be standardized. And also, critically, it highlighted the need for better physical chemical property characterization. So we had methods, but there was much more work to be done there. And more recently, in addressing this physical chemical characterization need, a report on fit-for-purpose testing and reporting guidance has been published by the OECD. And in these reports, it was stated that the characterization has to take into consideration basically the pre- and post-exposure scenarios, answering the questions such as what they are, where they go, and what they do. And also, because these are the things that will influence the end results. And then also there was a decision framework which included Solna principles was also suggested for material characterization, which basically suggests, asks to explain why this method has been used to characterize this particular property. So there is a lot of OECD work going on, and there are many different types of work objectives planned, which are addressing all the issues that I just spoke about. Most of them are in progress, but there is a lot of data as well. And if you would like to look at this data, you can always approach the OECD Secretariat, and they will be able to provide you the access to that data. And during this time, there was a very humongous surge in nano research activities, not just by the OECD, but also outside the OECD. And we did learn a lot from these activities as well. For example, it was discovered that nanomaterials, upon entering a biological or environmental space, acquire a new identity called protein or biocorona, which is defined by the type of biomolecules that it attracts. So a protein corona is suggested to guide the trajectory of a nanomaterial in a biological matrix or an environmental matrix, and it influences the reactive potential of a nanomaterial. So here the picture is very recent. One from a recent paper, and it shows in aqua blue and cyanine or green colors are the proteins that are bound to the particles, and in the blue fibrils are the functionalized nanoparticles. We also learned that there are many different ways by which these particles gain entry into cells for cells. And for example, we also came to know that the soluble particles exert Trojan horse effect. They get sequestered into lysosomal vesicles where they will get dissolved, and that creates an acidic environment, and then the toxicity ensues. And for fibers, for example, we learned that they induce a phenomena called frustrated phagocytosis. And as shown here in the picture, it simply means that a phagocyte trying to engulf a fiber is not able to do so because of its high aspect ratio as well as its rigidity, causing a frustrated phagocytosis leading to toxicity. So in all, I would say that there was a lot of learning here, and the examples that I'm showing here are basically from the inhalation toxicology, but I must say they all apply to other routes of exposure as well. And so the other critical aspects of routes of exposure were also noted, such as, for example, how the pH of the oral exposure route affects the particle dissolution, aggregation, etc. And even though the toxicity mechanisms are the same in oral exposure compared to inhalation exposure, the effects are different. And the main mechanisms of toxicity are basically oxidative stress, inflammation, and cytotoxicity and genotoxicity. So we do know a lot compared to what we knew a few years ago. With all this information, with all the data available, we still have no formal human health risk assessments conducted for any nanomaterials. Forgive me if I'm wrong about this. There are some attempts made by different organizations, but I'm not sure anyone has been completed to its successfully. So during this time, there was another realization, and that was in vivo animal testing is not a feasible option for nanomaterials, and we have to come up with alternatives. So to address this need, ODWPMN project was initiated, and the objective was to basically survey the state of the science in alternative testing strategies and to come up with a database of methods and alternative testing strategies used and for different materials, for different nanomaterials. And also a case study was conducted, particularly using about 641 data sets, the reports or the data sets from nano titanium dioxide studies to show like what are the endpoints in general investigated in this area in terms of the alternative toxicity testing, and how can they be used to support the risk assessment decisions. And at this same time, and the results were basically all that we could conclude was there was about more than 59% of the endpoints studied were associated with inflammation, genotoxicity, and cytotoxicity processes, the endpoints that we, the mechanisms that we highlighted in the previous slide as important for nanomaterial-induced toxicity. So there was another project at the same time, which concluded at the same time. And the same results that we saw in the OECD project were also highlighted in this study. And this study consisted of thousands of publications. And I think it spanned up until 2013, if I'm not wrong. And then later on, we added few more publications up until 2017. And this was also a part of another OECD project, an objective of which was similar, and that was to identify key biological events required to induce toxicity by nanomaterials. In this study, we had in vitro and in vivo endpoints separately investigated, and the literature was actually reviewed, and thousands of publications were used to conduct a word computing exercise, and then came up with the most enriched terms in all of these thousands of publications. And both OECD projects, regardless of the approach taken, the number of studies included, they both identified the same three endpoints, that is inflammation, genotoxicity, and cytotoxicity, to be the most investigated endpoints. One of the very important things that I want to highlight here is that the discrepancy between what we measure in vitro versus what we measure in vivo. And so in vitro, we are targeting some of the basic mechanisms, whereas in vivo, we don't know how these basic mechanisms are connecting to the in vivo endpoints that we have tested or we have reported. Most of this work is explained or described in the three reports that are here, OECD reports that you can take a look at. But the other important highlight of these studies was that regardless of how much information is there, the quality of this published data was very poor in the context of quality of material characterization details provided in these manuscripts, and also quality experimental design. And most of these studies lacked doses for the dose response analysis. There were insufficient variants of nanomaterials included. Therefore, we could not come up with a dose response matrix, or we could not come up with the property-specific effects that we were hoping to derive from this vast majority of the papers. And this might explain why we do not yet have risk assessment conducted on these materials. So this is the general overview of where the current state of science stays. But I'm going to switch gears here and quickly show you a couple of studies that we have done at Health Canada and how we have progressed from using the alternative or non-standard models and methodology to come to an in vitro strategy for screening nanomaterials. So this was my second nano study. I started at Health Canada in 2009. And the objective of my work was to basically understand the mechanisms of nanomaterial-induced toxicity. And I primarily used genomics tools and techniques that are not standard. And the other objective was to optimize non-standard methodologies and endpoints and approaches for inclusion in decision-making. So here in this study, we used the omics tools and identified a lung-specific acute phase reaction and identified a single acute phase response gene marker, serum amyloid A, which was sensitive to exposure and very specific to mouse lungs, which we were able to use and show what I'm going to suggest here is that it was sensitive enough to pick up the subtle differences between the different nanomaterials that we tested. As you see here, on the y-axis is the surface area, on the y-axis is the mRNA levels, and the x-axis is the surface area, and the different colors are the different nanomaterials. And as you can see, as the surface area increases, you are also seeing the higher expression levels of SA. And again, the SA3 mRNA levels were predictive sensitive enough to show the potency of materials to induce lung inflammation. And this is again shown here by the number of neutrophils that increase with the SA mRNA expression. So why is this important? The importance here is that the acute phase response is basically known to be activated in liver, but here we were showing that it is specific to lung as well and can be activated in lung. And two, this implied is that because acute phase response is known to be involved in cardiovascular diseases, prolonged and persistent expression of acute phase response in lungs could lead to systemic effects and raise the risk of cardiovascular diseases. And this work is now moving forward, and my collaborators in Denmark are using this information to come up with an adverse outcome pathway and come up with testing strategies for systemic effects of nanomaterials. And the next one is that so we kept seeing this inflammation and acute phase response in all of our studies in mice exposed to different types of nanomaterials. And over the 6 years, we had accumulated genomics data for more than about 30 or 40 different individual nanomaterials, and using the same microarray platform, same mouse of strain at the same doses and exposed at the same duration of time. And so it was a very robust genomics data, but we didn't know, because of the short protocols, we did not know what was the end adverse outcome here. And so what we did is that we decided to conduct a meta-analysis of all the genomics data that we had. So we pulled in-house generated genomics data along with the publicly available genomics data for nanomaterials, as well as the genomics data that is available for the mouse disease models, lung disease models. And lung diseases that we included in this analysis were like infections, inflammation, fibrosis, emphysema, allergy, etc. So the results, what you see here is that, and also the 3 different particle types were included: titanium, multi-wall carbon nanotubes, and carbon black. What we see here is that the titanium dioxide particles, even though they had inflammation and acute phase response, they did not cluster with any of the disease models. Whereas the carbon black was in the cusp between the nanotitanium and CNT cluster here, suggesting that it is toxic, but not as toxic as carbon nanotube, of course, depending on the endpoint we are looking at here. And carbon nanotubes had very interesting profile here. And the 24-hour post-exposure samples clustered with the bleomycin injury model and bacterial infection model, basically highlighting the type of inflammation these nanotubes were producing. And 28 days samples clustered with the something called Th2 response model, which is an adaptive immune response model that we see in fibrosis disease. So the whole experiment showed us that the underlying mechanism of carbon nanotubes induce disease. And what was also interesting is that it looked pretty much like a bleomycin-induced fibrosis. So using this information and digging a little bit more into the literature, we came up with an adverse outcome pathway for lung fibrosis, highlighting the key essential biological events involved in this disease process. For those of you who are new to the concept of adverse outcome pathway, it is simply a simplified, modular, or linear representation of a complex disease mechanism using a series of key biological events occurring after exposure to a stressor at different levels of biological organization, which are essential to produce toxic effects. So, and then later on, we also show that this fibrotic mechanism is not only specific to carbon nanotubes, but it also is applicable to other chemicals, non-nanomaterial chemicals such as bleomycin, silica, metal dust, radiation, pathogens, etc., etc. So it's a generalized mechanism for lung fibrosis that we have here, and we identified the key endpoints that we can now target for measurement using in vitro methodologies. So I'm going to skip these two next slides in the interest of time. So once we had this AOP and key endpoints identified, we use this data set that is the in-house genomics, publicly available genomics for nanomaterials, as well as publicly available genomics data for all kinds of diseases. This time, not only lung diseases, but also other diseases and the cancer diseases, etc. Everything was included. About 50 or 75 publicly available data sets were included in this statistical analysis. And then we came up with a 17-gene pro-fibrotic signature that was effectively able to predict if a data set is pro-fibrotic or non-fibrotic. And we validated this 17-gene pro-fibrotic signature using a limited data set that we had. However, we were not able to conduct in vivo whole animal exposure to evaluate its trigger in the lungs of mice after exposure to nanomaterials because of the amount of resources needed. So instead, we optimized an ex vivo precision cut lung slice method, and which involves basically culturing of a mouse lung slice externally outside the mouse in a petri. So this method is called like a reductionist method, and the idea here is one mouse can yield about 20 viable slices, which is good enough for investigating one particle, including control and 3 doses and 5 technical replicates for one post-exposure time point. So using just 3 mice, we can complete investigation of one particle as opposed to using 20 whole mice for one particle in the real scenario. And plus this model maintains its original anatomical structure, and all cell types except cells that are recruited from outside the lungs are present here. And this is extremely important for the fibrotic disease process because it involves multiple cell types at different stages of the disease process. And the slices also allow us to conduct histopathological investigation, which is very crucial endpoint for regulators to evaluate this disease occurrence. So we showed that the combo of the gene panel, which is the 17-gene pro-fibrotic panel that spans the genes associated with multiple key events that we identified in the AOP in the previous slide, and along with the PCL technique, give us kind of a 21st century tool for the assessment of nanomaterial-induced lung responses. This is work in progress. We are trying to pre-validate this methodology and move it forward at the international level. So what I have shown so far is for how to go about testing raw nanomaterials using non-standard methodology and still be able to arrive with some information for weight of evidence, for screening, for prioritization, etc. But what if a nanomaterial is embedded in a matrix, such as the food matrix or the plastic or something like that? This is what we did. This is my last slide, actually. So here, the question here is different. Before even we start looking at the toxicity of these matrix embedded nanomaterials, one has to confirm the identity of the nanomaterial that is embedded in a matrix. And then we should be able to track it after exposure to the cells or tissues to say that, yes, it is present there, and then be able to associate that with the effects. So our Consumer Product Safety Directorate at Health Canada wanted us to come up with a method where we can screen some of the consumer products that are suggested to contain nanomaterials for their for the presence of nanomaterials, the type of nanomaterials, and also exposure characterization of these nanomaterials. Of course, we don't have such a methodology right now, which is fast and rapid and resource effective, but we use this so-called hyperspectral imaging microscope to screen really a large number of skincare products that were available on the Canadian market. So the premise of this the microscope is that under this dark field enhanced illumination, the particles in the nanoscale are brighter and shinier, as you are seeing them in this gold and silver and titanium squares. And each of these particles emits scatters light. Depending on the class of the nanomaterial, they may have different wavelength at which they scatter light. And that scattered light is spectrally graphed as you see them in inset. And as you see that the gold and the silver and the titanium, they are very different in their spectral graphs. And looking at the similarities and differences using a software which is called spectral angle mapping, then we can track the material that you have in the cells or in tissues after exposure to the care products or something like that. And what's important to note here is that this technique is only applicable when you know what you're looking for, but it does not work for an unknown nanomaterial. And so we had a very large reference libraries for very different types of nanomaterials that we have looked at in our lab. And so we were able to apply those reference nanomaterials and come up and identify some of particles in some of the consumer products and type of nanomaterials that are contained in these consumer products. And then we chose one anti-wrinkle face cream that had titanium dioxide in the nanoscale. I think it was an 8 nanometer titanium dioxide particle. And we applied it to mouse skin, and we were able to show here at the bottom right-hand corner particles penetrating into the several layers of dermis of the skin. And what was, hopeful was that there was not much that penetrated through the layers. And so it was very tiny fraction. And because of that, we did not go on to quantify. We were not able to quantify, and we did not try to look at the effects. But this is something that I think one can apply in the context of the food and matrix embedded nanoparticles that you are all going to discuss, I think, in the next couple of presentations. So with that, and here I guess it's already time, I'm going to conclude. We saw that there are many different issues and challenges, and they are all the same regardless of the route of exposure or the problems we are trying to solve here. And we also saw that the conventional animal reliance may not be a feasible option for nanomaterials, and we have to come up with different options, different strategies and approaches to look at nanomaterials and their toxicity. So moving forward, predictive toxicology may take precedence. And we still have the same problems that we had 15, 20 years ago, that we still need an inventory of well-characterized nanomaterials. We still need a fit-for-purpose physicochemical characterization, and we need to know mechanisms. We have to have the understand the nanobio interface, and more importantly, we need to have dose response, dosimetry, and toxicokinetics studies conducted, because we still lack a lot of knowledge in this area here. And finally, one day we have to be able to conduct human health risk assessment for these nanomaterials. With that, I would like to thank many of Health Canada students and postdocs who contributed to this work that I presented here, and my collaborators in the National Research Centre for the Working Environment in Denmark, Ulla and Håkan, and Health Canada for sustained funding. Thank you.
So my question pertains to the protein corona, and I'm wondering if the composition of the nanomaterial actually determines which proteins coat those nanoparticles. And also, what do we know about the protein coating in terms of how those particles are then recognized by the immune system and the inflammatory cells? So does that influence uptake and so forth?
Thank you. We almost lost you for a few seconds here. Yeah, protein corona is a kind of, it still remains a myth. We know that the proteins are attracted, and there is a soft and a hard protein corona. So the protein corona changes as it travels through different compartments of the biological organism or the environmental compartments. But to say that a particular coding or a particular chemical composition of nanomaterial would attract particular set of proteins, I don't think we have science there yet. I may be wrong again, but I don't think that we are there yet. And one of the things that we know is that it also depends on the abundance of these proteins in the biological milieu that the particle is in. And so most of the times we are finding these abundant proteins are getting tethered to the particles. And apart from that, I don't think I can say much about how the coding, like maybe the surface charges, that will help in attracting some different protein types and different biomolecules. But other than that, we do not know much.
Really quickly, and then I have another question online, and that is, when we move into these in vitro assays to assess toxicity of nanoparticles, it seems to me that the media in which these nanoparticles are actually being tested, we need to simulate the microenvironment in which they also exist in the animal. And so it seems to me that that's going to be a tricky way to assess. Are some of those considerations being made as you develop some of this technology?
Yeah, that is correct. And so we do use in these in vitro methodologies, like, for example, the type of work that we have conducted did not require to supplement anything, because we were using slices there where you have pretty much the lung environment, and we did the in vivo exposures. But in vitro, for sure, we use the simulated lung fluids as supplement that contains surfactants. And we know that the surfactant chemistry is very important for nanomaterial clearance, nanomaterial interaction with these epithelial cells in lungs, for example. And same goes with the gastric fluid. So people do use gastric fluid to understand how materials behave in that particular pH. So the pH of any biological matrix is very important in determining how these nanoparticles behave, and whether that pH, how that pH affects their aggregation and agglomeration status, and how that impacts the toxicity. So yes, there is a lot of consideration made, and I must mention about two of the European Commission nano research projects. One is called Smart Nanotox, and the other one is called Patrols, where they have come up with the particle dispersion methodology, specifically keeping in mind these variations in the pH of different biological fluids when exposing cells to nanoparticles.
We had one question online and has now disappeared. I don't know when you switch presentations if that affected that. Any other questions?
Towards the beginning of your presentation, you indicated that animal testing for nanoparticles is not feasible. But at the outset, you quoted from Rasmussen 2016 that some endpoints differ for nanoparticles. So if you didn't do the animal testing, how would you know the endpoints differ?
Yeah. No, what I meant is that moving forward, it's not feasible. Not because we can't conduct those exposures, it's simply because they are expensive and resource ineffective. So the wide variety of nanomaterials that we have that we need to assess and we need data in the immediate future, we may not be able to use these tedious animal exposure protocols to assess all of those nanomaterials. That's what I meant when I said it's not feasible. And most of our work comes from in vivo. Yes, we need the in vivo, we needed those in vivo exposures and data from in vivo to really come up with the endpoints that we would be looking at in vitro. Without that, it is not possible. But now that we have this understanding and knowledge, we may be able to slowly move away from animal testing and making use of these in the key biological events that we may be able to target using in vitro and in silico methodologies. Does that answer your question?
How would you validate? How would you validate the in vitro and in silico?
Yeah, that's a humongous question, isn't it? And so, this is where the caveat is. In vitro toxicology has always been there. We have always used in vitro toxicology to understand mechanisms, but we have never used it for the regulatory purposes because we have not shown the connection. Like I was saying, the disconnect between the in vitro and in vivo is so high that we may not be able to use the data that is available there for assessing in vivo responses. And our regulators are used to seeing the clinical manifestation of a disease in vivo, and to come away from that to using in vitro toxicology tools that are only predictive of that occurrence, but not necessarily showing you the manifestation of the disease is a humongous thing. And to answer your question of validation, that's a big job. And if we were to go by the conventional ways of benchmarking against the in vivo data, that will not work for nanotoxicology for the same reason, because we don't have enough in vivo data to benchmark against. So we have to come up with an alternate way of validating in vitro toxicology tools, and I don't know what is that alternative way. So is it possible that we can use in vitro endpoints and then conduct some in vitro experiments for a large variety of nanomaterials, varying in their properties, in their chemical class, etc., and then use that data to validate the predictive capacity of these in vitro tools to predict the in vivo occurrence of disease? So I agree with you that we don't have an answer about to the question how we can go about validating the in vitro toxicology tools. And this is something that the regulators have to come forward with to say what is acceptable in terms of their confidence or the trust in this in these toxicology tools.
I have one more question that came in online, and then we'll have to move on. So the question is, as titanium dioxide is used in direct contact food packaging and has a portion of particles in the nanoscale range, are you aware of specific toxicity concerns related to nanoscale titanium dioxide that may migrate into food?
Thank you for the question. I have not studied particles in the food context, but based on the studies that we have conducted using different types of nanomaterials, which were raw nanomaterials as well as the matrix embedded titanium dioxide nanoparticles, we haven't really seen an overt toxicity. What we have seen is inflammation in lungs, inflammatory mediators being expressed at the high levels in vitro, but we haven't seen cytotoxicity per se in vivo or in vitro following exposure to titanium dioxide nanoparticles unless we are using very high doses, as we have done in one of our papers. Recently, I didn't show these results because they are not published. We are also looking at the genotoxicity of titanium dioxide particles, and we are looking at 10 different titanium dioxide variants. And what we see again there is that some coating of the particles, some of modifications, the property variants of titanium dioxide particles, moderately, very, I don't even know if it's a moderate, it's very insignificant, but there is some increase in the comet tail that we observe, suggesting that there are some DNA breaks being created by some of these surface-coated titanium dioxide nanoparticles compared to the pristine types of titanium dioxide particles. But again, the results are very marginal. And so it's my belief that based on the work that we have done, we haven't really seen something to call as toxic following nano titanium dioxide exposure.
Well, thank you very much for that presentation. And we'll move on to the next speaker. So our next speaker is Raymond Brines, and he is a review chemist in the Division of Food Contact Substances, Office of Food Additive Safety at the Center for Food Safety and Applied Nutrition with FDA. And Dr. Brines is going to talk on safety evaluation of food contact substances containing nanomaterials.
And then good morning to everyone. I'm again Dr. Raymond Brines. I'm a review chemist in the Office of Food Additive Safety, Center for Food Safety and Applied Nutrition. And I'd like to thank the organizers of this symposium for this opportunity to share with you the safety evaluation of food contact substances containing nanomaterials at FDA. So as a disclaimer, I note that the opinions expressed in this presentation are mine and not FDA's or any other U.S. government organization. And any mention of trade names or specific products should not be considered as an endorsement. So to give you a brief overview of what I'll be talking about today. So first, I'll focus on the regulatory framework for the safety evaluation of food contact substances. Then I'll talk about FDA's perspective on the regulation of products involving nanotechnology. And then I'll give a specific example of a guidance document involving nanotechnology, and that is the guidance document on significant manufacturing process changes. And then I'll give an example, a case study, where we have a food contact substance containing a nanomaterial, and in this case, it's a reverse osmosis membrane containing a nanomaterial. So FDA is responsible for regulating a wide variety of consumer products, including food, drugs, biological products, medical devices, and cosmetics. And among these responsibilities, ensuring the safety of our nation's food supply is the most relevant to this presentation. And one of the many aspects of food safety that FDA is involved with is the safety of food ingredients and food contact substances, which I will collectively refer to as food substances throughout my talk. The Office of Food Additive Safety, or OFAS, reviews the safety of information for food ingredients and food contact substances. And below here are the list of the various food substances that are reviewed by our office. But for today's talk or presentation, the most relevant is the food contact substance, or also called indirect additives. So what is FDA's legal authority over food contact substances? The Federal Food, Drug, and Cosmetic Act is a set of laws passed by Congress in 1938, giving authority to FDA to oversee the safety of food, drugs, medical devices, and cosmetics. And one of the amendments of the Act is the 1958 Food Additives Amendment, which actually established the pre-market approval of new uses of food additives. And also it defined what a food additive is. But or more importantly, the amendment that's relevant to this presentation is the Food and Drug Administration Modernization Act, or FDAMA. And that's because it defined what the food contact substance is. And also it led to the establishment of the Food Contact Notification Program. It has the same standard of safety and standard of review as food additives. So how does FDA assess the safety of a food substance such as food contact substance? For food substances such as food contact substance, FDA uses a unifying standard of safety, and that is FDA defines safe or safety as a reasonable certainty in the minds of competent scientists that a substance is not harmful under the intended conditions of use. FDA's assessments are based on safety alone, unlike, for instance, for drugs, where they look at the risk profile and the benefit of the drug substance. Also, the information necessary to demonstrate the safety of food contact substance is dependent on the exposure, which is determined by the intended use. So first, I'd like to define what a food contact substance is before I discuss the Food Contact Notification Program. So the legal definition of the food contact substance is any substance intended for use as a component of materials used in manufacturing, packing, packaging, transporting, or holding food if such food is not intended to have any technical effect in such food. So examples of which are listed in this bubble on the left here. So it ranges from antimicrobial agents to stabilizers to colorants. And they can be components of food contact materials on the right here, which can either be food packaging or food processing. All right, so now that I've defined what a food contact substance is, I'd like now to discuss the food contact notification and what type of information is needed also in a food contact notification submission. But first, let's discuss, when an FCN is needed. So there are three scenarios. One is if the FCN or the FCS is not authorized for use, and that is typically if the FCS is not regulated under 21 Code of Federal Regulation for the intended use, or not authorized in effective FCNs. The second scenario is if the FCS is new. And the third scenario is if modifications were made to an existing FCN. For instance, if there's a new manufacturer, a new intended use, or a change in manufacture. So I highlighted the change in manufacture there, since I'll discuss that later on in my talk. And that's also relevant to nanotechnology, as I'll discuss later. But in terms of the review period, or how long does an FCN review take, we have a 100-day statutory review period. And typically, if there are no safety issues within that review period, then the FCN becomes effective at the 120th day date, and which means that the FCS can be used for the requested intended use. So what are the relevant information necessary to establish the safety of an FCS? So one is the chemistry information, and the other one is toxicology information. However, I also would like to note that there are administrative and environmental components into the FCN submission that are also important. But in the interest of time, I'll be just talking about the chemistry and toxicology information. So for the chemistry information, these elements listed here are what's needed in order to establish the safety from the chemistry perspective. So, well, what I'm going to do is I'm going to break these down into three different groups of information. So the first type of information is regarding the identity, and that actually addresses the what the FCS is, and also what the potential, what has the potential to migrate, meaning, what are the migrants. So based on these information, you can deduce what those migrants are. For example, the impurity profile would give you what the impurities are and what impurities may migrate to food, as well as the stability, which can give us some information about what the decomposition products are of the FCS and what decomposition products may migrate to food. So the next type of information is the migration levels in food. So this determines how much is migrating or how much will migrate to food. And this can be determined by migration studies or by performing or conducting calculations. And I know my colleague, Dr. Tim Duncan from FDA as well, he's going to be talking more about the migration study later on. But in terms of performing calculations, so one may assume that 100% of the FCS and its impurities may migrate to food and calculate the migration level from that assumption, or one can also do migration modeling experiments or calculations. So the last information type that I'll discuss is the exposure estimates. So this is calculated by taking the migration level and combining it with the packaging information. So the packaging information is actually related to the fraction of each type of food that may contact the article containing the FCS, and the fraction of the daily diet that's going to contact the food contact article containing the FCS. So the exposure estimates are usually expressed in terms of estimated daily intake in terms of like micrograms, typically micrograms per person per day. So once you know what the exposures are for the FCS and its impurities, then the exposures are then used and evaluated then we would evaluate if these exposures are actually supported by the toxicological data. So there are two types of toxicology information. The first one is safety narrative, which describes the safety basis of the safety determination. And the other one is the comprehensive tox profile, which lists all the unpublished and published safety studies. So FDA has a tiered testing recommendations for based on the calculated exposure values, and potential carcinogenicity of the FCS and its constituents are all assessed, at all exposures. So as you can see here, we have 4 tiers, and for example, for tiers 2 and 3 is where we would actually have need toxicological studies to support the cumulative exposure. When I say cumulative exposure, I mean that the exposures coming from all the authorized uses of the FCS are combined together. So the type of toxicological studies needed for tiers 2 and 3 include, for example, genetic toxicity studies and or subchronic oral toxicity tests. So FDA's safety assessments of FCS is based on evaluating consumer exposure to an FCS and ensuring that the probable exposures are supported by the available tox data. And once we have the exposures, we then compare the consumer exposure, in this case, the cumulative EDI, to the acceptable daily intake, or ADI. So the ADI is an estimate of the amount of FCS that may be consumed daily over a lifetime with a reasonable certainty of no harm. And this may be determined from the toxicological data as well. So if the cumulative EDI is less than the ADI, then that means that the FCS is safe. All right, so now that I've introduced to you the safety evaluation of an FCS, let me change gears and talk to you about the FDA's current thinking regarding the products of nanotechnology, including FCSs that contain nanomaterials. FDA's approach to the regulation of products of nanotechnology is best summarized by the article in Science, published in 2012 and written by the former FDA Commissioner, Dr. Margaret Hamburg. And I'd like to highlight the attributes from this paper. So the first one is maintain a product-focused, science-based regulatory policy. So FDA's technical assessments are product-specific and takes into account the effects of nanomaterials to the products and their intended use. The safety review follows that, I discussed earlier for food contact substances. The second one is legal standards for risk vary among products classes. So as you've heard earlier, I mentioned that for food contact substances, the safety of the food contact substances are considered safe based on a reasonable certainty of no harm from their intended use. And then the third one is when there is an existing pre-market review authority, FDA includes attention to whether the use of nanomaterials suggests the need for additional data on safety, efficacy, or other attributes. But when statutory authority does not provide for pre-market review, then FDA encourages industry to provide safety information to FDA before marketing the product. So the fourth one is technical advice and guidance to industry. So FDA will develop and has developed product-specific guidance documents related to the use of nanomaterials in FDA-regulated products. And this is to assist the industry to meet their regulatory and statutory obligations. And for example, I'll discuss later on in this presentation a specific guidance, which is on the significant manufacturing process change and its relevance for food substances, including food contact substances. And then the fifth one is continue post-market monitoring for products that contain nanomaterials, and then they should take actions as needed. And then it's industry's obligation to work with the current information in product development and also monitor products once they're marketed. Based on those attributes from our approach, FDA's approach to the regulation of products of nanotechnology, FDA has developed, like I mentioned earlier, several guidance documents to better explain our current thinking on nanotechnology. So one of those guidance documents is the guidance on considering whether an FDA-regulated product involves the application of nanotechnology. So this guidance document does not establish regulatory definitions of nanotechnology, nanomaterial, or nanoscale, or other related terms. Rather, this helps the industry when they should consider potential implications for regulatory status, safety, effectiveness, or public health impact that may arise with the application of nanotechnology in FDA-regulated products. So that said, the guidance document actually identifies 2 points to consider when evaluating whether an FDA-regulated product involves the application of nanotechnology. So the first one is focusing on dimension, and the other one is focusing on properties. And I'll show you in the next few slides what those specific points are. So the first point to consider is whether a material or end product is engineered to have at least one external dimension or have an internal or surface structure in the nanoscale range. So approximately 1 nanometer to 100 nanometers. And this is consistent with the definition of other organizations in terms of the nanoscale. The term engineered here is also distinguishes products that have been deliberately manipulated from those that are or that unintentionally contain materials in the nanoscale range. So the second point is whether a material or end product is engineered to exhibit properties or phenomena, including physical or chemical properties or biological effects that are attributable to its dimensions, even if these dimensions fall outside the nanoscale range and up to 1 micrometer. So this extends the range from 100 to 1,000 nanometers for the nanomaterials. So now let me talk about the a specific guidance for food ingredients and food contact substances that may involve nanotechnology. So this is what we call as the guidance document on the significant manufacturing process change, which was finalized in 2014. So this is based on the consideration that manufacturing process for a food substance may evolve over time, and some of these changes may be considered significant and may impact the safety and regulatory status of the substance. So this is why FDA developed this guidance. So this guidance outlines the considerations and recommendations for assessing the impact of a significant manufacturing process change on the safety and regulatory status of the food substance. What is considered a significant manufacturing process change? So I've listed, several examples here, but I would like to direct your attention to the last bullet because this is relevant to nanotechnology. So this is a change in manufacturing or ingredient technology, such as the use of emerging technology, like for example, nanotechnology, that affects the particle size distribution of a food substance. So you might wonder, what is the impact of the significant manufacturing process change to the safety or regulatory status of a food substance? Basically, a significant manufacturing process change may affect the identity of the food contact substance, for example, and that may affect specifically the physical and chemical properties of the substance, which can then affect the intended technical effect, self-limiting levels of use, dietary exposure, and safety studies. And these elements ultimately affect the safety of the substance, which or may render the food contact substance or food substance in general not within the scope of the of authorization or regulation. Nanotechnology is an emerging technology that can be used in food manufacturing. So FDA considers food manufacturing processes that involve nanotechnology in the same manner as other food manufacturing technology. The application of nanotechnology may result in product attributes that differ from those of conventionally manufactured products and may merit a particular examination. However, FDA does not categorically judge all products containing nanomaterials as intrinsically benign or harmful. So our safety assessments consider characteristics of the final product and the safety of its intended use. When a food substance is manufactured to include nanometer version of the food substance, then the safety assessment should be based on the data relevant to the nanometer version of the food substance. And if the nano-engineered substance has new properties, additional or alternative testing methods may be necessary to determine the safety of the food substance. For instance, particle size, surface area, aggregation, or shape may impact the ADME, or absorption, distribution, metabolism, and excretion of the food substance, and potentially the safety of that food substance. In such cases, it's necessary to further examine the effects of change in these properties, including effects on the bioavailability of the food substance and also the transport across the gut. So what are the recommendations for significant manufacturing changes? The first one is manufacturers should determine the changes made to the identity of the food substance resulting from the significant manufacturing process change. In particular, they should look into the changes in structure, properties, purity, and the impurity profile of the food substance. The second one is taking into account any impact of the change in the identity of the food substance. They should also conduct safety assessment for the use of the food substance and paying particular attention to the bioavailability and toxicity of that food substance. So the third one is depending on the type of food substance, they should consider the impact of significant manufacturing process change to the regulatory status of the food substance. And last, or second to the last, consult with us about their conclusions about the impact of the significant manufacturing change on the safety and also the regulatory status of the food substance. And lastly, if warranted, they should make an appropriate submission. So now I'd like to discuss a specific example involving a change in manufacturing process of food contact substance. In particular, this example involves a reverse osmosis membrane involving nanotechnology. So reverse osmosis membranes are regulated in 21 CFR 177.2550. And these membranes are intended for use in processing bulk quantities of liquid food to separate or permeate from food concentrate or in purifying water for food manufacturing. The regulation specifies the various types of formulations to manufacture the RO membranes, including cross-linked high molecular weight polyamides depicted on the left here, and also the cross-linked polyethylamine as shown on this slide, on the right side of the slide. So the regulation also specifies the regulated adjuvants, suitable sports and conditions of use for the various RO membranes. So visually, a typical RO membrane is consisted of a polysulfone support, as shown here on the top, and then on top of that is the RO or the resin. In this case, it's a polyamide resin. So what happens if you introduce nanomaterial into the RO material? So, like for example, this construct that Zhao et al. In 2012 studied or investigated. So they incorporated proteopolymersomes, which are biomimetic structures where proteins are embedded into an amphiphilic block copolymer bilayer, and these form spherical structures which are around 100 nanometer in size. So in this example, they used aquaporin Z as the protein, which acts as a selective water channel, and that improves the RO membrane properties of the substance. So how do we assess the safety of that novel RO membrane? So here I'm showing you the different factors that we look at to assess the safety of that novel RO membrane. So the first one is we have to determine if there is a change in the identity of the FCS. And in this case, like I mentioned earlier, the proteopolymersomes were introduced into the resin, and that ultimately changes the identity of the food contact substance because you introduce new substances in there. And also the size of those substances actually is within the nanoscale range. So that is going to be considered a significant manufacturing process change. The second factor that we consider is if there is a change in regulatory status of the FCS. So originally, like I've shown you earlier, the regulation for the reverse osmosis membrane specifies specific formulations for the various components of the RO membrane. And now that we've changed the identity of the resin, then that changes the identity of the FCS, which means that the novel RO membrane is not within the scope of the regulation anymore. The third consideration is conducting a safety assessment of the FCS. So in this case, we will use the same safety assessment protocol that we use for conventional FCSs. But then there are also other considerations needed for doing a safety assessment of the novel RO membrane. And for instance, we have to look at the unique attributes of the FCS. And for instance, in the publication that I showed you earlier, the study by Xiao et al., they mentioned in their publication that the polyamide resin actually acts as a protective layer for the proteopolymerosomes from the environmental elements. And that means or implies that the proteopolymerosomes are protected and may not be able to migrate into food. So with that consideration, we then evaluate the exposure to the FCS and its impurities. And given the totality of the information, it's the exposure to the FCS and its impurities are actually, or is, very low. So with that, I'd like to conclude my presentation. So I've discussed with you the FCN program, and I've mentioned that it's a pre-market authorization process by which FDA regulates FCS, and that also includes the FCSs containing nanomaterials. The safety assessment of the FCS involves information on the consumer exposure, which again is based on the chemistry information, and also the toxicological information. In terms of the FDA's perspective on nanotechnology or nanomaterials, I've mentioned that FDA uses some considerations for products containing nanomaterials, and that involves or focuses on the dimensions and properties of the substance. And lastly, for food ingredients and food contact substances, I discussed the guidance document on significant manufacturing process changes, and I've shown that it addresses the changes in size distribution of the food substance. And with that, I'd like to thank you for your time and attention, and I'll take any questions if there are any.
That is, in terms of the toxicology testing of nanomaterials, many of these are not water soluble, and with the greater emphasis on moving away from animal testing and moving into in vitro systems, this is really going to pose a lot of challenges. And I'm just curious, internally, the discussions you have within colleagues within FDA, how are we going to do this?
Yeah, if you look at the nanomaterials, you can see, a wide range of different types, and they also vary in terms of the solubility. But there are ones that are soluble in water and ones that are insoluble in water. But I think one of the things that we should consider is how it's presented in the gut. Like, for example, some of these nanomaterials are insoluble, and usually they agglomerate. So if we are going to study these things, we have to make sure that they represent the actual form of the nanomaterial when they are presented into the system. So those are considerations that I think we also talk about within FDA.
It's a really important point that you just made, and I hope we follow through on that. Great. And,
just to maybe add to the discussion, there are also models that are more physiologically relevant, 3D, around the face, things like that. So you can incorporate that into the in vitro model.
Great. All right. Well, thank you very much, Ray, for your presentation. We're a little bit behind schedule, so let's take 10 minutes and reconvene if we can. Thank you. All right, I think we should go ahead and get started. So it's my pleasure to introduce our next speaker, Dr. Timothy Duncan. So Dr. Duncan has been with the FDA since 2009, and his role at the FDA is a research scientist and primary investigator in the Division of Food Processing Science and Technology. Primarily there, he does studies on the health and environmental safety of nanotechnology-enabled food contact materials and develops nanosensors intended to improve the agency's ability to rapidly respond to foodborne disease outbreaks. And so it's my pleasure to turn over the microphone to Dr. Duncan. The title of his presentation is The Migration of Engineered Nanomaterials Out of Nanotechnology-Enabled Polymers and into Foods.
Okay, thank you very much. It's my real pleasure to be here today, at least in the virtual space. I'm really sorry that I am unable to be there in person. I think that my colleague Ray gave a really excellent overview of the way that FDA treats, well, food contact materials in general, but in particular, the way it views packaging that may incorporate nanomaterials. I'm going to offer a complementary view into the technical aspects of migration of nanomaterials. I do like to point out that FDA does a lot of research, and our goal as research scientists is to provide the programmatic offices, like Ray's office, with key scientific knowledge and data that they can then use to make important regulatory decisions. So I'm going to start the talk by giving a brief overview of migration of conventional substances, because I think that it serves as important context to some of the challenges that nanotechnology presents to our understanding of migration. So let's start with what is migration. So migration is the transfer of a food contact substance from the packaging material to food under a certain set of intended use conditions. And so this is a fundamentally diffusion-based process driven by a concentration gradient. So you have a high concentration of your food contact substance in the in this case, a polymer, and a low concentration in the liquid, or the food, which is often a liquid, which drives the mass transfer from one phase to the other. It's mediated by the relative solubility of the substance in the polymer and the liquid, but fundamentally is a entropic diffusion-based process. So as Ray mentioned, it's incumbent upon a manufacturer to provide, among other things, a migration estimate as part of a safety assessment. So there are a lot of ways a manufacturer can go about doing that. Probably the most straightforward and easy way is just to assume that all of the substance migrates. That's certainly an easy thing to do, but it's often undesirable because it can lead to a vast overestimation of migration. And so it is frequently more desirable to either experimentally measure the migration or do a theoretical calculation. So when it comes to measuring migration, it's a conceptually straightforward process. Basically, you would take a portion of your food contact material and cut it into sections. Would string it onto a wire as shown in that diagram on the bottom left of the slide, and you stick it into what is often called an FDA migration cell, which is basically a glass vessel. You would fill it with a food simulating substance, and I will talk about that a little later in the talk. You would store it then under a certain set of time temperature conditions and remove aliquots at key time points and measure that simulant for the presence of the food contact substance using a validated analytical techniques, often LCMS or GCMS for a small organic molecule. If you do that at a lot of different time points, you would generate a curve that looks something like shown in that plot. I have a certain one time point highlighted. That's a 10-day time point. So, so for instance, for a room temperature application, it would be a common time temperature test condition would be 40 degrees for 10 days. That's an accelerated experiment. It's hard to do migration experiments for 6 months at room temperature. So there is sufficient data to show that if you do this at 10 days for 40 degrees Celsius, that's approximately equivalent to 6 months at room temperature. So that you would measure it at that time point, and that would be your reported migration level. FDA, as Ray mentioned, the FDA publishes a large number of guidance documents that lay out the agency's current recommendations for, in this case, best the way that the agency views is a recommended way to measure migration. So what I have shown down at the bottom of the slide there is an example life cycle process for a packaging material. So the packaging material is created, then for a food, it would be processed usually in some way, often under high temperature or high pressure. Then that material would be stored under a variety of potential conditions, either refrigerated or room temperature or in the freezer. And then often a consumer will take that package out and then heat it in either a microwave or possibly an oven. So depending on what the intended use of that condition is, you would want to measure the migration under conditions that provide a realistic realistic but conservative estimate of the exposure. So what this guidance document does is it says, well, if the intended use is, say, to store it for room temperature and then heat in the microwave, this is the condition that we recommend that you measure the migration. And so, there will be a certain amount of time at a certain temperature with a certain food simulant. And so this is all laid out in that document. It's a publicly available document, and it's really important to note that these recommendations are based on a couple decades worth of experimental data that's published in the literature. So these aren't just pulled out of a hat. They're really a data-based recommendations. And most of those studies, of course, are done with a conventional, what I call conventional migrants, usually small molecules in polyolefins. In addition to the potential to measure migration, you can also estimate it. I only have one slide on estimation, but since migration is fundamentally a diffusion-based process, you can use the diffusion equation to predict the migration if you know a certain number of key factors, such as the relative solubility of the migrant in the polymer in the food phase, and in particular the diffusion coefficient. So a lot of predictive models are really based on predicting a diffusion coefficient. So this slide shows one of the more common diffusion models that is used. It's called the Peeringer diffusion model. Basically, it really only involves a couple parameters. You have your molecular weight, you have your temperature, and then you have what's called a matrix-specific or polymer parameter, this AP value. Again, this is a value that you could look up. These have been generated over several decades generated from several decades worth of experimental data. So you can put those values in there and get a diffusion coefficient, which then you can put into the diffusion equation and estimate the migration. So what I have shown down there is a plot of the Peeringer model. That's the blue line with an AP value of 11.5, which is the literature value for LDPE at like say room temperature. And then I have a bunch of experimental data points that I took out of the literature for some potential migrants. And one thing I want to highlight is that the predicted line for diffusion coefficient is always, the predicted diffusion coefficient is always larger than the experimental data points. And that's, you want that because you always want to over-predict. You want it to be realistic, but you always want to over-predict the migration. You don't want to under-predict the migration. And so this is by design. There is a continual refinement of these AP values. There are a lot of people who just accumulate more data to refine these, particularly for, say, newer polymers for which there isn't as much data. Or, if you were to subject the polymer to a new type of processing condition or something of that nature, you might want to generate new AP values. So that, again, you have a realistic but conservative prediction of the diffusion constant. Okay, so that sort of provides a basic overview of migration of organic molecules. But what I want to point out here is that the migrant, in this case, for a small molecule, usually doesn't change during migration. This really makes an analyst's job a lot easier because generally an analyst will know what they're looking for. This also means that because it's usually, in the past, has usually been a small molecule, you really only need one analytical technique to quantify it. Usually, like I said, LCMS or GCMS, which doesn't mean it's an easy analysis, but it does mean that it is well within the skill set of pretty much any analytical chemist. And as far as prediction goes, you really only need to know a couple factors, such as the molecular weight, the relative solubility in the two phases, which is fairly easy to predict, and some polymer data for which there is generally a substantial amount of experimental data accumulated for. So the reason I bring this up is that it's a fairly straightforward process at this point. The migration physics is really well understood, and so it's generally a fairly straightforward process to either estimate or predict migration of conventional food contact substances. Our research really so one of the main research goals of our packaging group is to understand how emerging technologies may or may not challenge this well-established model. And so my particular interest is in polymer nanocomposites. So it's worth briefly describing what a polymer nanocomposite is. So a nanocomposite is if you take some sort of host material, most commonly a polymer, and you disperse into it some sort of nanoscale filler. It's some material that has at least one, often two or three dimensions on the nanoscale. These can have all different types of compositions, things like nanoclays or carbonaceous materials, bio-based materials, and a whole range of inorganic particles such as metallic particles or metal oxide particles. In the food packaging space, there has been a substantial interest in using nanocomposites. These are some examples that have been presented in the peer-reviewed literature over the last decade and a half. These are not necessarily authorized for use in the United States, but as I said, there has been a substantial amount of interest in some of these applications, which include things like UV blockers, antimicrobials, sensing applications, processing aids, anti-counterfeiting inks, a whole range of functional materials, and also some high barrier plastics. So just as with a conventional food contact material, if a manufacturer would want to incorporate a nanoparticle into a polymer that's going to contact food, they would have to use that process that Ray outlined in his talk, which includes a determination of the migration or the transfer of that nanoparticle or other substances from the polymer into the food. Unlike the conventional model that I've already presented, nanomaterials present some complexity that you don't see with a small molecule. And that complexity derives from the fact that there are a number of different mechanisms through which one could envision the transfer of these materials between the two phases. So, of course, we have a diffusion-based process, which would be the analogous process to the diffusion of a small molecule across the interfacial boundary. But we could also have things like a direct desorption of particles that are incidentally bound at the interface, or a dissolution of the particles, which in this case would be the particles themselves aren't really moving around, but you could have, say, oxidative chemistry on the surface of those particles and then transport of those oxidation products into the food matrix. In addition, we have what I often call post-migration or post-release processes, whereby, say, diffused particles can dissolve, or dissolved particles can reconstitute into whole particles, or particles can change shape, they can change size, they can aggregate, and so on and so forth. And it's really important to get a handle on this complexity, because each of these potential migrated forms could have different toxicological or pharmacological endpoints. Nanomaterials also introduced a number of challenging questions, such as how do you uniquely identify a migrated nanomaterial? What experiments do you need to identify and quantify the migrant? If you need to distinguish between, say, particle and ionic forms, you can't just use a single technique necessarily anymore to both quantify and speciate. And how do you predict? Some of the predictive models that I presented before, they're basically molecular weight based. What's the molecular weight of a nanomaterial? So you can't really use some of these established models to do prediction. So these are some of the research questions that we set about trying to answer. It's worth noting that a lot of research has been done on previously on nanoparticle release from polymers, both in a food context and also in a context of other types of consumer products. I have presented just a really small sampling of some of these research studies in this slide. I don't really want to go through them individually. The point of this is to show you that they've been performed on a range of different types of materials, some of them commercially available, some of them in-house manufactured. In the case of commercially available materials, it's not often really known what is actually in them or how they're made. Some of these are proprietary materials. And so what you see if you take like the 35,000-foot view of this body of literature is that the migration levels kind of are all over the place. Some studies use just total migration measurements. Some of them try to speciate. So some of them are reporting, just like if for instance in the case of silver, maybe total silver. Some of them are reporting particle versus ion. I think it's uncharitable to call it a mess, but it does. But the data are sort of all over the place, which makes it very difficult to get at fundamental migration mechanisms. And that information is really important to both understand the physics of the process, but also make appropriate recommendations for how one ought to measure the migration from these sorts of materials. So when it came to us trying to decide how to provide a useful entryway into this research area, we decided that we wanted to take a more fundamental model system-based approach. And so what I mean by that is that rather than using a commercial material, maybe we can step back and smartly design test materials that are intended to answer specific structure function questions. So this slide just captures that model system approach using one of the earlier studies that we published. In this case, we wanted to answer a simple question as if you put a smaller particle into a polymer, do you get more or less or maybe the same amount of migration? And so we answered that question by using semiconductor quantum dots. These are small particles. Most people are familiar with them in the context of television sets because of their fluorescent properties, and the fluorescence color is tightly tied to their size. They're also commercially available in fairly tightly controlled size regimes. So we figured that this was an ideal choice for a model system to study this question of the relationship between size and migration. So that's step one of our approach, is to intelligently design a system intended to answer a specific question. Number two is that we manufactured our own materials in-house. We don't rely on commercial manufacturers to produce these. We believe that by taking this approach, we know exactly how we're making them and we know exactly what we're putting into them. So we have our own processing equipment in our laboratory where we manufacture, in most cases, thin films. We then characterize the materials. So in this case, we looked at the fluorescence properties of these. So you can see in that photograph that we don't lose the fluorescence properties when we put these into films. The original idea was that we could use the fluorescence properties to track where the particles go and also some of the chemistry that happens on their surfaces. We also use advanced characterization techniques to understand, for instance, the way that particles are distributed throughout these materials. And then we do, of course, our migration or release experiments. So that little figure on the bottom left shows some of the release data from this study. The green bars represent smaller quantum dots and the red bars represent larger quantum dots. And so it's fairly apparent in this case that the smaller quantum dots give more migration. So we can start to understand, by doing this holistic approach, we can start to maybe connect some of the structural or chemical properties of particles versus their potential to migrate into, in this case, a food system. We did a lot more with that quantum dot system and continue to do some stuff with that, but I'd really like to focus the rest of the talk on some of our research on silver particles. For many of you know that silver, there's been a lot of interest in potentially using silver as an antimicrobial material, although it's not currently authorized for that use in the United States. Globally, there has been a huge amount of interest in this. Silver, in addition to that, that food context, these are also serve as good model nanoparticles because, first of all, they are fairly easy to make with mild conditions in a lot of different sizes and with a lot of different surface chemistries. They are also highly redox active. So one of the things I didn't mention with our quantum dot system is that we determined with some other experiments that the primary mechanism seemed to be the oxidative dissolution. So it's that dissolution-based mechanism. And so because the oxidative chemistry on the surface of polymer embedded particles seems to drive the migration, we wanted to use a highly redox active particle as a good model system. So in our first study with silver, one of the first things we wanted to look at again was this question of size, and so we made particles with lots of different sizes in our lab and incorporated them into low-density polyethylene. Image of one of those materials is shown in the top right-hand side of the slide, and then of course we did the release experiment. As with the quantum dot system, we do observe an inverse relationship between particle size and release. And in particular, it really becomes pronounced for particles less than 10 nanometers in size. Once you get larger than that, it seems like other factors start to become more influential on the release properties. We do, and you can see the difference between 4 nanometer particles and 10 nanometer particles is really large, a couple orders of magnitude. And we've also done this same type of experiment with our quantum dot based system, and we've started to actually use this large amount of data, including kinetic data, to begin to build predictive models. None of that is published yet, so I'm not going to present any of it today, but I do want to just kind of use this as an opportunity to show you that by this model system approach where we can really incorporate good quality particles into polymers, we can accumulate really meaningful data and start to understand some of the fundamental physics and chemistry underlying this process. Again, if we look at the actual the food simulant, we did this in a dilute acetic acid and into water, and we always see more release into the acetic acid, which is consistent with this idea of dissolution. Acetic acid is, of course, a more aggressive reactive substance to the surface of polymer embedded particles, and this is pretty consistent in the literature. In addition to the size, we can use this silver this silver system to look at maybe the effect of composition of the embedded particles. So we studied metallic silver particles. We also studied silver sulfide particles and embedded gold particles. The idea here was that if we change the composition of the particles, we can change the nature of the chemistry that happens on their surfaces. So if we compare the metallic silver particles with the silver sulfide particles, we see significantly more release or migration of the metallic silver particles versus silver sulfide particles. It's a couple orders of magnitude difference. The reason being that silver sulfide particles are already in an oxidized state, and silver sulfide is a really stable substance. So the composition certainly matters, and this is a significant result because both of these are fundamentally silver particles. So if you're creating a silver-based material, it's really important to know what the form that silver particle is in when you disperse it into a polymer, because that clearly has downstream implications on the potential exposure. Gold particles, as you might expect, are highly resistant to oxidation, and that's reflected in the migration behavior. We see almost no migration out of silver or gold composites, even when the particle size is really, really small. And also, it's clear that oxygen really drives this process. It is an oxidative chemistry, surface chemistry, and so if we do the same experiment under an argon purge night atmosphere, we see very little migration. So again, this approach allows us to really start to understand the chemistry that happens on the surface of polymer embedded particles. We often think of, I think it was initially thought that if you take these and put them into polymer, the polymer protects the particles from any sort of chemistry, but that clearly isn't the case, that chemistry really drives the migration process. I'd like to spend the rest of the talk talking about a study that we published this year. And to begin that, I'd like to present a little bit more detail this idea of using food simulants. Food simulants, if you wanted to measure migration, of course, you could use a food, right? So if you're intending to use your food contact material in tomato soup, you could do the migration in tomato soup. But food presents a lot of analytical challenges. It has a lot of interference, it's complex, and so it's pretty much standard practice at this point to use a food simulant. So a food simulant is basically a simple solvent that it mimics the chemical properties of certain categories of food, and these are spelled out in that chemistry guidance document that I mentioned at the beginning of the talk. This slide just captures a small selection of the recommendations. Certainly, you should contact that guidance document for more detail. But for instance, if you wanted to, if your intended use was with an aqueous food, you might use a water or a 10% ethanol. If your packaging was for an alcoholic substance, you would use an alcohol-based stimulant. You might use a food oil for a fatty food, and so on and so forth. If you look at the migration literature for nanomaterials or nanocomposites, most of the studies use food simulants, and they tend to use two of them in particular, usually a 3% acetic acid and an aqueous and water. It certainly makes sense why they do this. It's consistent with the current recommendations, both in the United States and in other countries. And so this is pretty much standard practice in the nanocomposite release literature. But particularly as we've shown that nanoparticles and nanomaterials have significantly more complex chemistry than what you might see in more conventional substances, it was fairly surprising that researchers used these simulants without really asking the question as to whether they were appropriate to use or whether they by using them, you would get an accurate measurement of the potential migration. And so we really wanted to focus on this question in a little more detail. And the hypothesis of the study was motivated by the following. So we know we have, if we take a material that has, say, a silver particle in it, we've already shown that, say, into acetic acid, the primary migration is going to be, in this case, silver ions, represented by M+ for a generic metal particle, but let's say silver ions. If we were to use a acetic acid stimulant, this would probably be an accurate picture. However, we know that foods are complex mixtures. They include all kinds of different chemicals in them, including emulsifiers and antioxidants and lipids and inorganic substances and dyes and sweeteners. And a lot of these substances are redox active. So we hypothesized that if you had a redox active substance in there and your metal ion was also redox active, you could undergo redox chemistry in your food and basically re-reduce those migrated metal ions back into, in this case, silver particles. There's actually a substantial reason to believe this because there have been a number of reports of using sweeteners deliberately for green synthesis of nanoparticles in the literature. Now, those were fairly those studies were directed specifically at nanoparticle synthesis, so they added things like capping agents and high concentrations of sweeteners and things sugars and so forth. But it stood to reason that if people can do this deliberately, that it might actually happen unintentionally in a food system. So we focused this first study on sweeteners, the reason being that sweeteners, and by sweeteners I'm really talking about sugars, many sugars are potent reducing agents, and so you seem to have all the ingredients here to facilitate this process. And so the hypothesis of our study was that if you do have migration of silver ions and you do have a sugar present, that you could attenuate the identity of what the final migration product would be. And so the first step of this study was essentially to survey a bunch of different sugars and sweeteners and determine whether they could reduce silver ions into silver particles. We initially set out with a high concentration of silver. Again, there's no packaging material here. This is just to take a look at whether this is was a viable hypothesis. So we took a look at a lot of these single ingredient solutions. We also took a look at some commercial beverages, which although they are simple foods, certainly they are still quite a bit more complex than a single ingredient solution. So, you know, they have things like, in some cases, organic acids in them. They might have colorants. They can have a fruit pulp in one case, and so we wanted to look at a few of those. We also looked at some milks with different fat content. Of course, milk has a lot of sugar in the form of lactose, and then we looked at a couple of wet foods, including a yogurt and a starch slurry. Because this was with a high concentration of silver, certainly more than you would see migrating from a packaging material, we're able to use just the color changes as a analytical tool to monitor silver particle formation in these test solutions. And you can see here these are just photographs of the vials. So basically, we just took solutions of these substances and added a silver ion, and then just took pictures at different times. And going up and down are the different time points, and left to right are the different ingredient solutions. And you can see pretty much we always get color formation, with one or two exceptions, which is showing us that in most cases we are getting silver particle formation under these conditions. We also see it in the wet foods. So there on the top right, you can see yogurt and starch. They both form silver nanoparticles over time. Starch was an interesting case because starch is not a reducing ingredient, so just the slow hydrolysis of the starch into sugars is enough to produce silver particles. And of course, we also get it in yogurt, which is a little interesting because yogurt has a lot of fat. It's also viscous and acidic. So even under those conditions, which would seem to be unfavorable for silver particle formation, we do get silver particle formation. I think that we are the first people ever to show silver particle formation with yogurt, so that was kind of interesting. In the paper, we do take a really close look at some of the differences in the silver particle formation of these different substances to really better understand some of the chemistry that goes on. I don't have time to present that all today, but I do want to just note two things. One is in the red box. You can see that a lot of these have a dark purple color. This is indicating that in a lot of cases the particles are aggregating. Again, we haven't specifically added any capping agent or anything here, so that sort of makes sense. But it does show that if this were to be happening in a food, in contact with a food contact material, aggregation is something that you might have to be tracking. In the blue box, I highlight the fact that even in most cases we do see color gradually appearing over time. In almost every case, we also see it eventually disappearing over time. This is indicative of a redissolution process. So you have particle formation due to reduction of silver ions, but then you have subsequent dissolution of those particles, probably due to the presence of oxygen. So the final substance that a person might be exposed to is really going to be determined by a balance of these two competing kinetic processes, which are very different depending on what the nature of the formulation of the product. So, you know, if you have something in there that can stabilize particles, you might disfavor that redissolution process. But if you have an acidic substance, that can really favor the redissolution, and so it's really sensitive to the food formulation. We used electron microscopy to actually take a look at what we were forming. So we get all different shapes and sizes of these particles. We get hexagons, we get donuts, we get kind of flower shapes, we get triangles, we get star-shaped patterns. It again really is sensitive to what the ingredient formulation is. And we can track this also in time with electron microscopy. So not surprisingly, we can see that they do grow over time, and probably if we waited long enough, we would start to see them dissolve again. So that was at a really high silver ion concentration. We also wanted to look at a silver ion concentration that was more more realistic as far as a food packaging application would be. In this case, we can't actually look at color changes because these are so dilute, so we turn to single particle ICP-MS. A single particle ICP-MS, for those in the audience who may not be aware, is an analytical technique that has found wide use in nanoparticle analysis. It is basically like regular ICP-MS, except rather than averaging the signal over a long period of time, we instead monitor the signal in real time. And the idea here is that if you have a particle going through your plasma, rather than getting a constant signal, you get a big burst. And the size of that burst is proportional to the size of the particle. And so by measuring the signal in time, you get all these spikes, and the statistical distribution of the peak intensity can be correlated to the particle size distribution and particle number concentration. So it's a really useful technique for studying in this case, particle formation in the presence of some of these food ingredients. So this is some sample data. This is done at 0.25 ppb silver. So this is, again, a concentration that is more akin to what you would see migrating in a food contact use situation. In the presence of a sugar, domino sugar at a concentration about what you would see in a soda, stored for 10 days at 40 degrees C, and you can see that it's very clear that we get spikes in our single particle ICP-MS trace, which is showing us that even at this low silver ion concentration, we are getting silver particle formation. We can then translate that data into particle concentrations on the left and median size distributions on the right. Again, there's a whole kind of a mess of trends here that I'm not going to go into, but I do want to highlight one set, which is our domino sugar set. And in both cases of the particle concentration and the size, we are seeing that the particles do seem to grow and then redissolve. So everything that we see here at the low silver ion concentration is consistent with what we saw at the high silver ion concentration. So all those same processes are relevant when the silver ion concentration is more at a level that you would see migrating from a food contact material. Finally, we wanted to actually see if this happens with a silver nanoparticle, a polymer that's dispersed with silver particles. So we manufactured, again, our own particles. We incorporated them. We phase transferred them so that they're compatible with the hydrophobic polymer domain, and then incorporated them into LDPE. You can see some photographs of those at the bottom, and then use them to manufacture these little miniature sachets or packages, which we filled with our sugar-containing simulants. We looked at three simulants in this case: just a water, which is shown in blue, a squirt, which is a citrus-based soda, shown in red, and then the domino sugar solution shown in green. Just first draw your attention to two things here. If you look at the green line versus the blue line, particularly at 15 days, it's immediately apparent that whether we're quantifying by the particulate mass, which is panel A, or the particulate number, which is panel B, or the size, which is panel C, we are getting significantly more particle formation in the presence of sugar than in water. So this is showing us, confirming our hypothesis that if you have silver ions migrating out of these materials, in the presence of sugar, sugar can reduce those particles back into, reduce those ions back into particles. The squirt, which is shown in red, is an interesting counterpart. You can see we actually have more particles formed in squirt at 10 days, which then are significantly less at 15 days. So again, this is this process. Squirt is acidic. It's a soft drink, and so as opposed to domino sugar, where the particle reduction or the ion reduction is the process that wins out, in squirt, ultimately the dissolution process wins out, and so. The distribution of the exposure distribution of particles versus ions is again really sensitive to what the formulation is, and also what the time point or the storage conditions are. Finally, when we started this particular project, we fully expected or predicted that the presence of sugars would really only affect the balance of particles versus ions that would be in the migrate solution. We didn't really think the sugar would affect the total amount of migration. In fact, we were very wrong. We actually found that not only does the sugar reduce silver ions back into silver particles in the food simulant, it also seems to draw out a lot more silver. We don't really have a watertight explanation for this. we have hypothesized that it is due to the fact that particles, as they are being formed, actually act as an effective sink for silver ions, so that you maintain a low concentration of silver ions in your simulant, which maintains a high concentration gradient across the polymer interface. So you maintain a high driving force for migration. That's our current basically hypothesis for that. But the idea here is that the exposure, the total silver exposure, is going to be in a food that contains sugar. The total silver ion migration seems to be, based on this small study, to be a lot larger than you would determine if you were to use just a water-based simulant. So obviously, this is a small amount of data. This is something that we want to look at into a lot more in a lot more detail, and we've also spent some time in the last year looking at some other potential redox active ingredients that might commonly be found in food, and we've also found some interesting things there. So it's an issue that we continue to look at. We think it is merits further exploration. So with the last minute or two, my final slide, I just want to take a step back again and review what we have learned so far. So we started out with this idea of, which I call the molecular migration paradigm. This is the analogy to what you would what we know is the case for small molecules, where you have whole particles basically diffusing out of the package and into food. And at this point, we know this is not an accurate picture. Rather, migration tends to be driven by oxidative chemistry at more or less stationary particles. We leave that orange dash line there because there is maybe a potential under some conditions for particularly really small particles to diffuse, but we regard this as a minor process at best. The oxidation of the particles leads to formation of metal ions, which are much more easily transported out of the packaging material into the food, into the contacted food, and so the primary exposure was, we thought, going to be ionic. There is a role of the acid. I didn't have time to talk about that today, but it is a really active component in driving that chemistry, and particularly the ability of the packaging material to act as a kind of gatekeeper to what is allowed to go into the interior of that material and interact with those polymer-bound particles. So that's another really interesting area. Again, I don't have time to talk about that today, but it is a factor that needs to be taken into consideration. But now we've kind of determined that really isn't an accurate picture either. So the current paradigm that I'm working under is this reconstituted nanoparticle migration paradigm, where you do have this oxidative chemistry that happens with the particles and the transport of those metal ions out of the polymer and into the surrounding environment, but then also oxidative or transformative chemistry that continues to go on after the migration process is formally completed. So again, you have oxidation or reduction of those metal ions into particles. You can have reoxidation or dissolution of the particles after they're formed. It should be noted that those particles that are formed can have different compositions. So they may not be pure silver particles at that point. They could be silver chloride particles or silver oxide particles or silver sulfide particles. That's another thing that needs to be looked at in more detail. And then those particles can undergo other processes like aggregation or sedimentation and so on. This is a paradigm that, again, needs further exploration. I don't consider it completed at any point. As I noted, food systems are incredibly complex and diverse, and so any of those ingredients could potentially interact with particles and drive a transformative chemistry. So that's really what I have to present today. It's where we are at now with this research. I do want to offer a few acknowledgments to other scientists within the agency, particularly our packaging group here in Chicago, as well as some of the more regulatory scientists in the agency that we work closely with, like Ray, who presented earlier, to ensure that the questions that we're trying to answer are useful to those offices and can derive regulatory decisions. We also leverage a lot of students with the Illinois Institute of Technology to do this work, and of course our collaborators from other government organizations and universities. So I certainly thank you for your attention, and I'm happy to take any questions now or later.
And thank you very much for that interesting presentation. We do have a number of questions that have come in from folks listening online. And so let me start with the first one. The first question is, you mentioned that nanoparticles in food packaging are not yet allowed, but graphene is used in barrier water bottles already. Can you please clarify your statement?
I don't have any really comment on that. I don't really keep track, to be honest, as to what substances the program offices are authorizing for use and not. I don't know if Ray has a comment on that. And instead, yeah. So I apologize for that.
Move on to the next one. The next question is, are you recommending new migration studies for sugary foods and food contact materials for regulatory authorizations?
I don't make any recommendations. Basically, my job is to provide data and information to the program offices that they can then consider and decide whether either new guidance would need to be made or just new recommendations that they would convey to manufacturers if manufacturers consulted with them. So that's certainly not my role. I don't really know what affects that process as to how they change their recommendations. I think that the data that I have presented today is certainly suggestive of the fact that that the chemistry of nanoparticles is really complex, and we don't fully understand it. It is important to underscore, though, that this is a single study. I'm fairly confident, and I think Ray would probably agree with me that recommendations aren't going to be made without a substantial amount of data and reproducibility. And so I regard this as an exploratory study that is suggestive of the fact that it is something that we would want to look into further and acquire a lot more data. Once a substantial amount of data is made, if that data shows that changes in the recommendations are necessary, those type of changes would be made by the programmatic offices.
Okay, another one. Do silver ions that migrate from conventional silver-containing packaging materials lead to the formation of silver nanoparticles in beverages, or are these results specific to nano-silver food contact materials?
That's an excellent question. It is something that we have explored this year. I haven't really, to be honest, had a chance to fully analyze that data, so I don't want to tell you the conclusions of that before I've done that. But I mean, it certainly stands to reason. I mean, there's nothing about silver particles per se that would make them any more of a unique source of silver ions than a, I don't know what you might call a non-nano. I mean, silver is silver, right? The chemistry is more or less the same. It's silver 0 going to silver plus. And now, certainly, the kinetics of those processes can change, but again, that is something that is a question that we have thought to ask. It's something that we have looked at this year. I haven't analyzed that data fully enough that I can make a conclusion today.
And I just have one last question. So, one of the things I noticed as you were talking about nanoparticle release from these polymers, you didn't really talk about temperature. And should we just assume that, typically chemical reactions increase with temperature? Is it reasonable to assume that at higher temperatures you would have a faster release and more release? And again, I'm thinking about, as various products are transported around the country and the temperature that they may actually incur during transport, either cold or, in some parts of the country it could be very warm. I'm just curious if you could comment on that.
Sure.
Is there some general principles you could convey around that?
Sure. Okay. Yeah. Basically, so for the most part, we have followed the FDA guidance for a room temperature storage application. So we've done most of these at 40 degrees Celsius, always trying to keep that 10-day time point among our time points. We have done these types of tests. I'm trying to think if we did it with silver or not. I can't say whether we've done it with silver or not, but we have looked at the effect of temperature on some of our migration studies, and we do not surprisingly find that if you increase the temperature, you get more migration. That's consistent with basically every other chemical reaction known to existence. I should point out that it's not just the chemistry that's influenced by temperature, however. Certainly, since this is driven by a chemical reaction on the particle surfaces, you would expect those rate constants to increase as a function of temperature, depending on whatever the activation energy is. We don't have good information for those rate constants, I should point out. Even in solution, those rate constants are hard to determine. You can't just use bulk silver because surfaces of particles are often, they have more surface area, but the surfaces can also be a lot more reactive because of their curvature and also the way that those surfaces are modified. So even in solution, we don't have a good measurements for those rate constants. I certainly don't have any data that I'm aware of for rate constants for polymer-bound particles. But fundamentally, the chemistry is going to be very, very similar in the sense that as the temperature goes up, those rate constants are going to change. However, also the temperature influences the diffusion process because diffusion is dependent on the movement of polymer chains and the formation of void volumes, and that can be easily modeled by an activation energy as well, which is a temperature dependent. So the temperature dependence of migration of nanoparticles is going to be dependent on temperature. We've seen it dependent on temperature in at least one or two of the systems that we've looked at, but I don't have enough temperature data to yet to really understand the physics of that or model how the temperature affects in a quantitative sense that. And also, temperature is also going to affect that reverse chemistry of reforming particles back into reforming ions back into particles, right? So really temperature affects a lot of these different processes. So how does temperature affect what the final migration endpoint is? That's going to be a really complex question that's going to require a lot of data to study.
It's like from your answer, that would be the case. Yeah. All right. Well, thank you very much for that presentation, Tim.
Thank you for the opportunity.
All right. Our next speaker is Dr. Maria Rubino. And so Dr. Rubino is a professor in the School of Packaging at Michigan State University. She has also spent about 15 years in industry as a scientist for chemical and food companies, and she's a chemist by training. And one of the areas that she has been very much involved in her own laboratory is development of active surfaces using nanotechnology for applications in active packaging. And the title of her presentation, which will be in person, is Including Functional Nanoparticles for the Implementation of Active Surfaces. So with that, I'll let you take, and let me give you this.
Okay, thank you. Can you hear me okay? Good. Okay, well, thank you very much. I'm delighted to be here. It has been very interesting. Actually, it will be this presentation will align very much with the previous presentation because I will be talking also about mass transfer. So we have, why do we want to add nanoclay to packaging materials? And in many ways, it will improve the characteristics of the material. So it will improve the mechanical properties. It will minimize the use of polymer material because we will need much less material, much less polymer. And also, it will be very important how we produce that composite, because it's critical that those clays are going to be exfoliated and converted into this nanoclay. That is what it will give all these different characteristics. So when we add nanoclay into the packaging, we obtain, we have these silicate minerals, and we have the clay that we see in the in here, and we could see that there it's very much layered, right? It has a lot of layers, and so when we add it into the polymer and we process the polymer, that layers will open up. So we will see that the clay. Here is, what do we have in the clay? Well, we have this structure, okay, that is the nanoclay, have silicon, aluminum, and then we have an exchangeable cations here that are very important because it's what will facilitate the clay that is the surfactant that will allow now the clay to be spreaded into the polymer. So we have many components here that are very interesting, and we are adding into the polymer. And as when we add these component, these nanoclays into this clay into the polymer, we begin to extrude the polymer and stretch the film, we are going to be converting the clay into nanoclay because one of the dimensions is in the nanoscale. So we will see that although they are very, very large, these nanoclays are very large, they are very thin and they have one dimension in the nanoscale. And so this will help us minimize the amount of polymer. The polymer will have much more strength and for very small amount of material. But what is happening is that we are concerned about what is the fate of the nanoclay? And so is the nanoclay going to migrate when it's in contact with the food product? So that was the main question. So, but we have here the shape of the clay will be in the form of these platelets that are very in the nanoscale, and the surface area of the nanoclay will increase significantly. And all these, the nanoclay we have found when we were doing the literature review that they have some concern about toxicity. Are these nanoclay that are in the film, if it migrates, it will get into the food product, or because we are considering the application in the food product, and so these particles could migrate, and then this could be toxic. We didn't know, although in the literature there is a lot of concerns at that time of some degree of toxicity because they could go through different in the organism. So what is happening is that, and this is what we were thinking that could be, that could happen is we have the film that contains the nanoclays, and the nanoclays the nanoclays are here in the film, and we put in contact with the solvent that could be water or some other solvents, and eventually there is a migration of the moisture or whatever specific component is in contact the film. It will penetrate into the film, and some of the surfactant will migrate, or some of the nanoclay will continue to be released. Now, we don't know if the nanoclay, what is the state of the nanoclay at that point. So, so we need to, so what we were thinking is that if this material, this packaging material, and the nanoclay that is in the film will migrate, it could get into the food system, or it could get into the soil, or it could get into the water. And so this could be exposed, there will be an exposure. And this was all our hypothesis at the time, that these nanoclays in the could migrate into food systems, and so then it could be consumed. And we have some references in different papers that establish that there is degree of toxicity. So so we wanted to understand, are the nanoclays that are released from the nanocomposite into the different environments, how much is released and how is released? And so what are the physical and chemical characteristics of the nanoclay before and after the release process? So we decided, before we continue talking and looking at application of nanoclay, are they really being released from the film? And so we produced some nanocomposite, and we took the clay and we label it, and so we label it and with rhodamine, and we sure that put it in the film, and then do a migration study, and we observe and we track if there was some nanoclay in the solvent. So at that time, we observed that there was some of the nanoclay in the solvent. So we decided to produce film, polypropylene, and we used nylon, and we added nanoclay. Specific amounts that were recommended, and we use some compatibilizer when it is in the polypropylene, and we make the composite, and then we extrude the material, and then we have the film produced, and we characterize the film. We did an XRD to be able to see where all the different characteristics, and we look at all the results, all the thermal properties, the structure, and the morphology of the film. Here we could see this is the XRD for the polypropylene. The exfoliation is okay. Here is nylon, where the exfoliation is much better. So the dispersion, due to the nature of the polymer, is more compatible the clay in this case. And so we have a nice film, and then we did migration study, very similar to what Tim was discussing. We put the films. These are all different discs of film, and we put it in a solvent, and we look. We use in this case this instrumental analysis, graphite furnace analysis, graphite furnace atomic absorption, to be able to determine how much film in function of time was migrating. And we observe that the release of the nanoclay was much more in polypropylene, was higher in polypropylene than the release that was released from nylon, which we were expecting that. We use 3 different temperatures. It's very important when we do this release study. This part is where we will get all the information regarding the diffusion coefficient, and so this was an interesting information for us to understand what is happening, what is the fate of these nanoclays when they are in the film. So we continue, and the next step, because there is another component in the nanoclay that facilitate the clay to be dispersed into the film, and it's the surfactant. There is a surfactant that is added, and so we wanted to know if that surfactant also migrate, because that wouldn't be it wouldn't be a good thing either to have too much surfactant, because this is coming into the food, right? So we have exactly the same system, except that now we will have different. We have a liquid chromatography with mass spec to do the detection, and so we detect how much of that surfactant was migrating, and we find that it was significant, the amount of surfactant migrating too. So at this point in this, we have developed a lot of metrology tools for the quantification of the nanoparticles that are migrating. We have characterized the nanoparticles in situ too. We have characterized, quantify and model the release of the nanoparticles from different components, from different matrices. So this, we realize that at this point, we know that the release of the nanoclay is in the low ppb levels. So the release of the surfactant is in the ppm level, so that is also important. And we have developed throughout this process a lot of different tools that will help us to talk more about, to do more analysis about and understand how is this migration process. So we decided at this point that instead of, maybe this is an opportunity to look because at what other application these nanoparticles could have with the film. So we thought that the engineering nanoparticles, like nanoclay, could be used for doing other type of activities too. We can implement active surfaces for innovative functional packaging materials, or packaging can be used as a strategy to improve safety and other functions for application, different type of food, pharmaceutical and chemical. So we decided that, well, maybe we could use an active surfaces and use the nanoclay as a vehicle to have the opportunity to look at add some other activity to the package system. So we can either take the nanoparticles and absorb it in the film. And in the nanoclay will have the active ingredient. It could be either absorbed, it could be attached into the nanoclay, and so then the nanoclay will be added to the surface of the film and become an active surface. So we mix these nanoclays with different type of active ingredients, and now we will have an active surface. We could also, instead of adsorb it, we could do it covalently. We could attach the active ingredient, okay, to the nanoclay, and then apply in a coating the nanoclay, functionalized nanoclay, and then put it in the film, and now the film become an active surface. So this is an approach that will create, by using coatings and applications of nanoclays, functionalized nanoclay, an active surface to protect the package. So the functionalized nanoclay could be used to tailor active surfaces depending on a specific application. We could use, we could develop novel surfaces and have the possibility to have a film that have a bactericide effect, or we have a film that could release some of the components of active ingredients, or it could be either attached or released. Functionalized nanoclay by grafting or absorption of the molecule with a specific activity. It could be an antioxidant, it could be a bactericide, it could be a scavenger. Incorporation of functionalized nanoparticles in the coating for the development of the active surface. So the clay will become the vehicle, and we will attach or absorb some of such active ingredients. So now we can immobilize we have different modes of actions. As I mentioned, immobilize the active ingredient and become a self-disinfecting surface. The absorption, if the absorption of the active ingredient is chosen, then the active ingredient could be released. The application will dictate the mode of action to be used. So we did some proof of concept about that, and we have different bactericide like ampicillin, hexanal, and nicene. We took these bactericides and we attach it, okay, to different, like this is the ampicillin. We attach it to the nanoclay, or we can absorb it into the galleries of the clay. So we characterize after we included the different active ingredients, and we quantify how much of the bactericides were included. And we tested using put it in this Petri dish, and so we add here is the clay alone, and then here is the clay with the active ingredient, and we could see that here the ampicillin in this case is really killing the microorganism in this, and this is after a certain time also the same thing. Here in the second line, we could see also the ampicillin, the OMT alone, and the OMT functionalized, and here is ampicillin alone, right? So here we have another image of, and we could see we use hexanal that we added into the clay, and so we could see here how the hexanal was behaving and against Listeria in this case and Salmonella. Also, we use grafting of the ampicillin, as I mentioned before, and we use some other approaches, and we could see here in this case we have the ampicillin, we attach it to the clay, and we have a functionalized material that will be added into the specific composition. We can also graft methine and into the clay, and then the grafted methine will put in the polymer or put it in the coating in the surface, and will act in order to prevent as an action. So coating formulations and application. The coating was formulated, and we functionalized the clay with different active ingredients. Coating with functionalized nanoclay was applied on surface of the polypropylene film in very small coatings of 0.5 mil. And the coating were also cured with UV at 365 nanometers. And in here you could see, here we have some examples of this is the plane, the control, and here we have added to the coating the hexanal, and here again different amounts, and we could see that the action of the functionalized clay. So currently we are working in developing more active surfaces and tailor with several modes of action. We immobilize, and the two ways are to immobilize by grafting the active ingredient into the clay, or to be absorbed within the clay. And the active surface application will be active packaging. We could use another application, could be done in mulch films, and also in equipment surfaces. This was an effort that we work with Rafael Lauras, and these were the graduate students. This was a PhD student and a postdoc and a PhD student that we have working in this project. And these were the fundings that we have for doing this work. So, comment.
Question? I have one. In terms of using the active surface applications of nanomaterials in food packaging, is this still all kind of laboratory-based, or is that actually being used now commercially anywhere in the world?
No, this is is not used yet. We are still developing. We are, but the advantage of this approach is that we have minimized the amount of bactericide that we are using, and we can use it in direct contact. And that is an advantage, significant advantage, because you don't use, usually one of the strategies was to add bactericides in the film and then they migrate, right? Here we are added in the coating, and it's very small amount, and it's directly where it's needed. So that is an advantage, and it's minimizing the amount of bactericide that is going to be in the actual product. So that is a very good advantage.
Seems like there's a lot of applications for this active surface applications, I guess.
Yes. Whoop. Yes, definitely it is very.
Yeah, just actricidal activity.
Right.
There's a lot of other ways that this technology can be used.
So yes, it could be implemented in many different applications. We did it for food safety because it was for us very important to get that route for the application. Yeah.
Okay, anyone else? Questions? If not, I think we're close to being out of time. We'll have lunch in the next room over. We have breakfast, and we'll reconvene at 1 o'clock. And so I'm pleased to introduce today Katja Hantel, who is a Senior Director of Sustainability for ConAgra Brands, where she works to integrate sustainability across the company's portfolio of leading frozen food, snacks, and grocery brands such as Healthy Choice, Orville Redenbacher, and I hope I get this right, Fornterra. She has spent more than 15 years developing sustainable business strategies and communications for global organizations in the for and non-profit sector. And just kind of as an interesting aside, she's also author of a book on U.S. media coverage of climate change as part of the St. James Studies in World Affairs series from Academic Press. So with that, the title of her presentation today is How to Talk About Science When No One Is Listening.
Thanks, Norb. I'm thrilled to be here today. And science communications is always a really important topic, but it feels especially relevant when we're living through a global pandemic and are exposed to science communications every day. Some are good, some are effective, some maybe not so effective. So what I'm going to cover is a summary of some science communications research as well as practical experience from the world of corporate sustainability communications. We'll talk about what's not working, so where's the gap, and then how to fix it. I'll give you some practical communications planning tips, and we'll talk through a case study that some of you may even be familiar with if you've enjoyed our Swiss Miss hot cocoa recently. So there is no shortage of science communications research and support. Here at MSU, elsewhere, the National Academies has a whole research agenda on communicating science effectively. My area of study and expertise is communicating environmental sustainability science to the public. So what I'll bring today is some research that I did at Duke, some of the great work also coming out of Yale and George Mason University, plus nearly a decades of corporate communications experience, working with the organizations you see here on this slide to communicate their environmental sustainability science to the public. So let's start with what's not working and use climate change as an example. The Intergovernmental Panel on Climate Change has been studying and communicating science for a very long time. They have put out more than 40 assessments and publications over 30 years. They have run major public communications campaigns. And yet, public awareness of these communications among US adults remains pretty low. Only about a quarter of the 27,000 consumers surveyed have been receiving these messages with any frequency in the media. And the public understanding of the science that IPCC and others are trying to communicate over these decades and through all these messages remains relatively low and relatively the same over the past decade or more. So this was my area of research. And what I found is that there's a science communications gap that applies not only to climate change, but many other fields of research and study around sciences. And there's also a way to build a bridge to fix it. One of my favorite science communications constructs comes from Yale. It is the idea that you take your research and you boil it down into 5 facts and 10 words. So 30 years of IPCC climate change research, they have boiled down, scientists agree, it's real, it's us, it's bad, but there's hope. So how do you do this with whatever your research is? Let's go through some practical tips and then a case study. Bridging the communications gap is really a 3-step communications planning process, but it is counterintuitive because it requires you to think about what you want to say last. First, we think about the audience. What's our desired outcome? What do I want my audience to think or to do? Second, what baseline am I working with? What does my audience currently know, feel, believe? And then what messages can I develop? What do I have to communicate that is most relevant to bridging this gap? So I'm going to tell you a story of a packaging life cycle environmental impact assessment as told to consumer media. Conagra Brands partnered with Berry Global, our packaging supplier, to develop a new packaging innovation for our Swiss Miss cocoa. We moved from a throwaway multi-material cylinder, which was the industry standard, to a lower carbon footprint recyclable plastic cube, industry first. So here's our data. Our packaging LCA key findings were around recyclability. So we found that about 40 to 60% of US consumers have access to curbside recycling programs that accept the new polypropylene container. On climate change, we found that that cube shape is stackable, meaning you can get more than 3 times the number of packaging units in a truckload, which reduces the number of truckloads we had to make to the filling station by 36, saving more than 1,000 gallons of diesel a year, which results in a smaller carbon footprint for that transportation part of the life cycle. 98 metric tons of CO2 emissions avoided, about a 15% reduction. And we found that these environmental benefits are generally applicable to other similar products in the portfolio, a 38 ounce or a 22 ounce. So how did we communicate this? First, we thought about the audience. The desired outcome was we wanted the media, the consumer media, to think ConAgra is innovative in sustainability, and we wanted them to promote the product message. Where are they starting from? Our audience knows and believes sustainability is important, but they aren't experts in the science and the metrics. Nearly 60% of consumers surveyed say it's important that the food products they purchase or consume are produced in an environmentally sustainable way. Yet more than 60% said they found it hard to know whether their food choices are environmentally sustainable. So we didn't run that survey, but it's illustrative of what we found when we looked at our audience. So what are our 5 facts and 10 words that bridge the gap? There on the left, you see the Yale construct. For Swiss Miss, this is an industry first. It's an eco-efficient shape. It's recyclable. It has a lower carbon footprint and more applications coming in more sizes. So 5 facts, 10 words is a foundation for content development. If you look at the news release we put out about this innovation, those facts are included in the headline through the first paragraph. Also understanding that our audience are not experts, we had to translate our data into their language. So coming up with relatable equivalencies for 98 metric tons of carbon. It's basically the same carbon footprint as driving from the Earth to the moon. And what were our results? So these are 5 real headlines, media headlines that reporters wrote based on our news release. And you can see that our 5 facts, 10 words are included in some of these headlines. So to summarize, we can build a bridge between scientific research that is data heavy and not really understandable broadly into science that can be understood, communicated, and promoted. 3 steps. First, think about your desired audience outcome. What do you want your audience to think, to do? Secondly, what baseline are they starting from? What does my audience currently know and believe? And third, then you craft your message. Can we get it down to 5 facts and 10 words? Thank you. I'm happy to take questions.
All right, any questions? I guess we were perfectly clear. Yeah, go.
So you went from what essentially was a paper-based product to a plastic product. Did you get any blowback from that? Did people question that decision?
In the media and the public?
Yeah.
No. Our message was recyclability, and our message was lower carbon footprint. And that really eliminated negativity around you're moving from a paper, metal to plastic. We did not receive negative feedback. Yes.
I would respectfully like you to reconsider the IPCC example that you used in terms of it's real. Because what that then connotes is that anyone who questions this becomes a climate denier. And the antithesis of science is saying that this is settled. And if you look, each year when the Nobel Prizes are awarded, Science magazine has a write-up on the individuals, in terms of their early research And in most cases, there's struggles to get these non-conventional ideas accepted. And how some talk about how they were kicked out or almost kicked out of their laboratory because they were promoting something so unconventional. And so I think that one should not try to portray science as dogma and something is cemented in stone.
Sure. I am presenting on science communications and how the IPCC could effectively communicate their findings. But I appreciate your point. Yes. Yes.
I just want to build on this question because I think it's important. And there is, in the media, I think there are narratives that are generated. And I think that there is kind of building on what he was saying is that there is suppression of alternative points of view. And so it begs, address the question, if a position that you need to bring forth rolls against the narrative and to the point, in fact, where a position you wish to present would maybe even be actively suppressed, how do we deal with that?
Should follow 5 facts and words.
Yeah,
How do you deal with that?
I mean, this is not a talk about how to navigate political science. I don't know how you navigate acquisition. Tell you how
I was just wondering if you kind of differentiate like scientific communication from either like public health communication or like public policy communication, because like in my mind, if I'm doing research and I have a result, it doesn't necessarily have a political, it doesn't tell you what a value judgment is, just tells you what the finding is. So how do you kind of differentiate between just communicating what the results are versus what value judgment you make and what policy you're advocating for?
You have a question?
All right, thank you. Yeah, all right, so. All right, does Oliver pronounce his name Jolliet? Olivier. Okay, you're going to introduce me or I'll introduce myself, I guess. All right, now do I need to turn this on? It's on. Hello, we can hear me? Okay, well, good afternoon. I'm Leon Bruner. I'm an adjunct professor here at Michigan State University and working with Norb Kaminsky on the CRISP program. And it's my pleasure to chair the session this afternoon. So I'd like to introduce our first speaker, Olivier Jolliet. He is a professor in life cycle impact and risk assessment in the Department of Environmental Health Sciences at the University of Michigan.
His teaching and research focuses on life cycle risks and benefits of chemicals and consumer products, foods, and emerging technologies. And he models and screens population exposures, intake fractions, pharmacokinetics, and so forth. So he studies a lot of topics that are of great interest to us in this area. He's given a lot back to the community. He's published over 200 papers. He's given lectures on life cycle analysis, and he's mentored 19 PhD students and 50 master's degree students. So he's got quite an accomplished career. The title of his presentation this afternoon is High Throughput Risk Screening of Chemicals in Packaging and Food Contact Materials. So thank you for being here.
Thank you very much.
Yep, go ahead.
Reach into your pocket.
That's good. Okay, and this is, okay, that's pretty cool. So to address and reach that, we have been working with an international team called USTOC. It's for UNEP CTAC Toxicity Assessment Program. And we first, in fact, worked mostly on the environment. But recently, we've really moved to ingredients in consumer product. And the goal was here to integrate exposure to chemical in consumer product in LCA, chemical alternative assessment, and high throughput screening for risk assessment. And we want a multi-compartment framework, which look both, if you like, we can look at from the product perspective, where we follow the chemical of the product, or we could, from a receptor perspective, one person is exposed to different product in that case, and differentiate, would say, an average user or kids from a high user population. And especially, we want to have both toxicity and exposure and hazard, and not stay on one side. Here is a team who has been developing that. It was coming from the life cycle assessment, but recently we had like Wei Shu from Texas A&M, who in terms of the toxicological modeling and dose response is very interesting, and colleague like Leo Postuma, who are more on the ecotox side here. So what is the framework? Basically, we want to start from the chemical mass in the product. So if I have a packaging and food packaging, what are the quantity of additive, for example, in a certain packaging, or the quantity of plasticizer? From there, we would like to determine, and I'll define it in a minute, the products. Oops. Page up. No. Okay. We would like to determine the product transfer fraction to our near-field environment, and eventually to the intake, so to know which fraction of this chemical mass will be taken in by the user or by different people. Use a dose response factor to either look at risk or to impact in eventually disability adjusted life years. We have to be conscious, it's orders of magnitude of impact in that case. So let's walk through that through an example, and first determining the mass of chemical in product and how it enter with the food packaging. So we have basically, we need the amount of product usage and the chemical content as a basis. So that would be my quantity here of, if I stay here, yeah, that's not good. You know where I am? It's not my presentation, so I move to the next presentation there. Sorry about that. Yeah, this is here. We're a bit higher than that. Yeah. Okay, thank you. So we start from the chemical in product, and we then look where does it enter. So USTOX has different points of entry which correspond to a different model. In our case, we would like to enter in a food contact material, which will be in contact with food, and from there look at the intake in either usually ingestion for a food contact material, but you could have the contact of the person with the packaging, and the packaging could eventually also lead to inhalation through volatilization in that case. Okay, I don't know what's going on. Let me, I think I'll manage. Okay. Thank you. So, for example, if I would take a packaging of LDPE, cooking bag for our rice down here, and it would contain like 0.9% of dibutyl phthalate. We found in certain database. I'm not sure if DBP is still used there presently. That would give us 31 milligram of DBP, which is entering my packaging. From there, we will look what are the transfer of the chemical in this packaging. So it could be directly transferred to the food and to the gastrointestinal tract, have a epidermal contact, or volatilize and then be here. Or at the end of life, it will be probably go to a landfill and could have emission, subsequent emission in this landfill. So the product intake fraction would be the fraction of what enter a human mouth, basically, or skin, divided by the initial mass in product that we have. And order of magnitude here was like 0.16%. In the case, it was the packaging would be heated with the rice inside it. So if you multiply your product intake fraction times the content, you will arrive to an intake here of like 0.05 milligram ingested per packaging in that case. Okay, so we have the same approach we can use for article interior in that case, which would be like building material or toys, cosmetic. We can do, of course, we can do. In fact, it's interesting because we started the first 10 years of our activity mostly on the direct emission to the environment with life cycle, and we've been a bit stupid because we should have started with a use phase. Okay, because that's where we are. We have, in fact, very often the highest exposure in most products, and so now we can do it. A cleaning product from object surface or paint when they are wet, some pesticide residue, and the food contact material program. So for food contact material, we know should not touch too many things. Okay, so we have here's the Piringer model that we know, and basically when we look what it does, it follows a diffusion here, which is a pure diffusion, which would go here to infinity, but at one point you saturate to a value, which is determined in fact by the partition coefficient here between the food and the packaging. And you have a sum of exponential, basically. And what is interesting is, in certain cases, when you start having a high KPF and a low alpha value, which is linked to here, you need to take a sum of like 50,000 exponential to be accurate. Because if you have even 100 exponential, and most people work with like 20 or nodes in that case, you might really overestimate by orders of magnitude what's going on. So the thought was, how could we simplify this model? And we published that a few years ago here. And basically, we take at the start, we take the diffusion, okay, which is linked to the diffusion coefficient. So what is driving the star of this curve is in fact the diffusion inside my food contact material. And then you will have a 2 exponential model. It's a bit subtle how we manage, but with 2 exponential, you could be accurate if you choose your coefficient in the right way. What is interesting is this model, we see we have really 2 main factors, which is the diffusion coefficient and this partition coefficient at the surface. So it's the ratio of the concentration at the surface of the packaging to the concentration in the food that we are considering. Okay, so how good was the model? This was measurements from FDA, and we see that the model is pretty good with an R-square here of 0.85 to predict the migrated fraction against the measured fraction here. This was when we had measured diffusion coefficient. Now, Alexei Anstov, who did that work, tried now with a simple diffusion coefficient equation, which was relying on molecular weight and just temperature. And you see that in that case, you're ruining a bit your regression because you have an R-square of 0.39. I mean, if your input data are not good, you are in trouble. Okay, so we worked after that to develop further and say we need better equation for the diffusion coefficient. And here we came out, it's a publication of 2017 here, where we see that we have the molecular weight and temperature, but also then a coefficient which is linked to the material itself. And you see that we are in log scale here, so 5 being a factor 100,000. So depending on the material, it's not only depending on the chemical. In fact, for this one, the molecular weight has a small influence, and it's mostly depending on the material you're considering. Here, HDPE being substantially higher than PET in that case. And with that, we can arrive for a prediction of the diffusion coefficient with a relatively good prediction. Still, you still have 1 order of magnitude here variation, which means also 1 or 2 orders of magnitude variation, which means that you also have 1 or 2 order of magnitude variation on the results. Okay, but look, the diffusion coefficient vary bit. These are we collected about 1,500 chemical material combination, and these vary by about 20 order of magnitude. Okay, in that case. What about the KPF, the partition coefficient? Here it depends on 2 things. It depends on how much the chemical likes oil, the log KOW, the octanol water partition coefficient, and it depends also on what the type of food we have. So in fact, we have the highest KPF. So high KPF, it means it likes the packaging versus the foods. And so with water, in fact, you will have a low migration rate, whereas with oil, you will have a low KPF, and that will increase the migration rate. The saturation will happen, in fact, at higher level in that case for that. And again, we could in fact have an explicit, we have even here an interaction term with the log KOW and the ETOH. The ETOH between, and we've seen that in the presentation of our colleague Tim this morning, who was showing the importance of the equivalency of the food. So ETOH of 0 is like water, and 1 would be pure alcohol in that case, or oil here. And again, we have a relatively good prediction here. The points here are in orange was not part of the training set, so we can predict really as prediction. What is interesting also is for the KPF, there is in fact the variation of this number vary only by 1 or 2 orders of magnitude. So KPF depend mostly on the KOW and the ETOH, little on the material. So different behavior depending on the different coefficient. And it's an example here that would be a sterilization where you have 30 minutes at 125 degree, a bit an extreme case in terms of, but to show the tendency. And you see that here, a PET would have a very low migration rate at this level, but much higher if you have HDPE. And you see the difference here between oil, milk, and HDPE with water, which changes here. So this different combination matters in that sense. I like this sort of graph. This is a heat map where we look here at the fraction migrated in log scale. So this is 0, 10%, 1 per 100, 1 per 1,000, 1 per 10,000, okay? And this is basically the saturation value, which is proportional to the inverse of the KPF. And here's the diffusion coefficient. So what is cool here is you can see that in that range, okay, you're not sensitive at all to alpha until you reach this diagonal here, where you're completely sensitive to alpha and you're not depending on the diffusion. So you can see, depending on the combination, where you stand, what is your material, you can know, in fact, which of the two coefficients you need to improve. And because one play a role in a certain range, and the other in another. So this is interesting. This is example here. I don't know this guy here. He doesn't have that's an additive of packaging. And you see that then the ETOH make it vary in that case, but it doesn't matter so much because it's in range. It would matter if we would be in that direction here. We would have large changes with the ETOH in that case. One cool thing is, in fact, we recently have been studying toys and mouthing of toys. In fact, the mouthing of toys can be approximated by a food packaging model where we take the saliva with, I think we took an ETOH of 50% in that case, as the food in contact or the saliva of the child in contact there. And you see that we are able with this model here to predict, these were measured migration rate, predicted migration rate against experimental migration rate, against, you see, across here 5 different material and the family of chemical, the phthalates leading to the highest migration rate here. Not necessarily that they migrate quicker, but they simply are in toys at high concentration. They are in the 5 to 40%, the plasticizer. So the content will be also important, and in addition, the fraction migrated in that case. And we can start doing high throughput risk application. And here we have not rerun. I mean, I'd be interested in a project where we would collect, in fact, content in material and systematically apply it for different material, different ETOH, and screen that completely. If there is an interest of Chris, I'll be ready to work on that. We have here for toys mouthing, we just published that 2 months ago. And you see that this is the hazard quotient. So I like this sort of graph where we have here the mouthing exposure. That's the microgram here per kilo per day. And we have a reference dose, which is a safe dose, and I've put it reverse. This is a small reference dose, high toxicity. This is a high reference dose, low toxicity, okay? And we see that here we are starting to exceed the hazard quotient of 1, not too much in a sense, and this is mostly the plasticizer, and this is due to their high exposure. It's not that the phthalate have a super high, in fact, impact in that case, but simply they are at higher doses here. Okay, so now how do we compare what we have that in different applications? So now I take a life cycle application, and we had been using that in the life cycle initiative, which is an initiative from the United Nations to improve the life cycle of products. And we took that as our basic case study. We could have taken other things, but we wanted to look also at the rice packaging in that case. And we look at the entire life cycle from my rice production, starting from my agriculture production, the paddy rice, the primary packaging here, point of sale, and finally the cooking, where we had a gas stove, but also an open fire with fine particulates. So we were looking at a broad range of impact in that case. So this was here the inventory of kilo emitted per functional unit. We are rather, I mean, we are here, functional unit would be 1 kilo of rice cooked, of white rice cooked in that case. And we see that the quantity, these are the emission to the outdoor environment, okay? And we have here, it's not emission here, it's a quantity inside the packaging that we will multiply it by my product intake fraction to get the quantity. So here we are in kilos, so the milligram would be about here. Then this is where we had our 30 milligram per kilo rice applied in this case. So now this was the exposure, and we can calculate our exposure. We need to compare with the toxicity. And here we've been developing inside the Life Cycle Initiative and this UN initiative a systematic way to first determine what would be a starting benchmark dose based on animal or human, and we are going to extend that with, in fact, in silico estimate afterwards. And it's interesting because we have regulatory data here for 744 chemicals, okay? And we have used a database from Richard Judson at US EPA, who has about 8,000 chronic experiment on 8,000 data, okay? But there are no regulatory data for 7,000 of them. So here we could go from 1,000 basically or 700 regulatory data to extrapolate for the other. And we see that the 25th percentile of the benchmark dose, which is an effect dose 10% or an OL in that case, is in fact very well correlated with regulatory data. So it's interesting that the regulatory data are not necessarily the most sensitive. I don't know if because new data have not been considered or they are not considered as the most substantial, but it enabled us now to move to 8,000 substances. And we've explored, I think with random forest algorithm, we could be able to predict that with R square around 0.5, 0.6, which is not great, but not necessarily much worse than an animal model or in vitro, in fact, extrapolation as an alternative. So applying that, we could arrive to an impact of these different chemicals which are environmentally mediated, and we can differentiate if it's via soil. And we were seeing that, in fact, the food packaging was in fact substantially higher than the impact. So it really matters compared to the life cycle emission. And when we were looking in more detail, it's interesting. Here you have the life cycle. These are human health impacts, and trying to evaluate in disability adjusted life years equivalent with, as I say, uncertainty factor is a 10 to 100. And you see the packaging itself is really relatively low compared to the impact. Here we have the cradle to grade rice. This is start. We have the fine particulate emission, which are playing an important role in that case. And the rice packaging is in fact dominant here. I'm not sure we have the cooking here. The indoor cooking might be separate. But we have here the rice packaging. No, it was not considered the indoor cooking because it's also really high in the same order of this one. And was in fact dominant in that case with what was considered. Now, additional question, how does this compare with the impact of the food? You were speaking I'm not interested in the packaging. I'm interested in what is inside my packaging itself. So recently you might have seen and heard about hot dogs. We published about basically the microdally gain or lost with 5,000 food items. But in the 10 words and 5 things, people have taken, we featured the hot dog as a nice example. It went viral. I mean, in fact, the potential reach has been 1.3 billion people. So in terms of communication, for good or bad reason, right? It went viral in that case. So, I mean, if you want to reach this paper, use this one. You don't need to pay. This is a free download that you don't find with a DOI in that case. So that was cool for once. I mean, to be the impression that perhaps people will think next time they buy. How do we calculate that? How do we calculate the 36 minutes? And what is the accuracy? Because it's not exactly probably 36 minutes. In addition, that's an average of our population and not a single person. But the idea was to say we took the global burden of disease, which is normally at population, and bring it at the level of the food item, knowing that we should look at the overall diet. But for most Americans, we are in the active range where we don't eat enough vegetables and we eat too much of processed meat and too much of red meat, for example. So take your hot dog, and your hot dog has, in fact, 61 grams of processed meat, some sodium, which is detrimental, some trans fat, and on the positive side, have a bit of fiber, that's my bun, polyunsaturated fat and calcium, which are positive. So take your 61 grams of processed meat, the global burden of disease, considering also that microdally, anybody has a sense of what a microdally is? Probably not really, but if you realize you have 500,000 minutes in a year, 1 per million of a year is in fact half a minute. Okay, so I can translate now in minutes gain or lost in that case. And I have the risk factor, and this is done for 15 different age group, the 2 gender and outcome specific for 50 outcomes. So there are 6,000 dietary risk factor. But as a whole, when we recombine them in a cumulative way, we arrive to about 0.5 minute loss per gram processed meat. We multiply that, and this is specific to diabetes and ischemic heart disease for the processed meat, where the calcium will act on colorectal cancer. And we account here also for the severity, how many DALI per case of these damage in that case. If you multiply it, you arrive to 61 times 0.45, 20 minutes loss just for the processed meat, another 7 minutes with the sodium, you lose another 3 minutes, so we are 37 minutes lost, and we gain 1 minute with the PUFA, the calcium here. So 37 minus 1, we arrive to 36. Uncertainty from about here, 22 to 45 minutes is the range of uncertainty using Monte Carlo analysis and the interval of the burden risk ratio from the burden of disease. We repeated that for 6,000 food items, including rice, okay? So I will not comment too much, but not to be too depressed, in that case, US American food diet is heavy on this one, okay? That's not good news for here. So you better to make like Asian food or Asian ethnicity diet is about 20 minutes of life gain per day compared to the average US. But you can we had the nut pecan pie. The pie was not perhaps the good part of it, but for the nuts, you can gain 25 minutes with 30 grams of, this is per serving, of nuts and fruit and vegetables you can do. So you can look at that. We have 160 food. We then look at the carbon footprint and 18 different impacts. But if we look now, my rice, if I compare white rice, I'm losing by eating it about 2.5 minutes. My my packaging was not negligible. It was a minute lost per, in that case, per serving. So this is for 140 grams here of rice. Now, if you instead of eating white rice, you eat brown rice. I don't know why it stay here. This is in fact 8 minutes gain in that case that we have because we have the whole grain which has this positive effect in that case. So I think since I've never dreamed that we could arrive to anything like that. And it's order of magnitude. I think the tendency are interesting in that case. Crucial that we are conscious and communicate also the uncertainty of that sort of results. So USTOX is able to cover the following product and type of, and I'll be interested to collaborate on that. So life cycle assessment, that's where we're coming from, and we have here some application on the food contact material. High throughput screening, we've published with US EPA a study on 5,000 chemical product combination, showing in fact the personal care product for the population might be really important, but the maintenance product and building material are really high for the user of this. So when you paint your house, use not a mask like that, but a real one. In that case, high throughput screening with like children toy, building material paint, we're publishing that. And the risk prioritization and the chemical alternatives assessment also, we've been using that. So if I conclude here, the migration model in USTOC enables to accurately and rapidly, that's only a 2 exponential calculation. We can even do that in Excel in that case. Calculate for various scenario and material. And that's the beauty to have this prediction for the diffusion and the KPF, which have an improved accuracy, but still need to be improved. So we see that the diffusion and partition coefficient are the one which are important. And via that, the temperature will play an important role, but it's really via the diffusion coefficient mostly. The new model have good performance when compared to measured both diffusion and migration coefficient. And we are in a cool time where we're working on the exposome, where we could start comparing a whole series of different exposure. And even I would say, not probably with food contact material, but if you take the NHANES study where we have the concentration in the blood of various chemicals, we can link that nowadays to the mortality data and start exploring what or which of these chemicals might have influence on the life expectancy of people. And this should be done with machine learning because we cannot just think of all the interaction in our small head. Okay, thank you very much.
And maybe I calculated lengthening or shorten your time of life. Right. Given an like a act, is that kind of what you're here?
Forever.
Well, I got that one.
Good. And, of 30 grams of nuts per day or 250 grams of fruit, there is no additional benefits. So that's where we have to look at the of how much reduction in cardiovascular disease. Mm-hmm. I mean, the limitation is apple.
Sure.
Because this I still like to counteract, but they're not necessarily on the same type.
Yeah. And I guess the other part is duration. So here is my
is that what we've shown is two things: processed meat and beef. Okay.
I can't do without beef.
Health, the beef is crucial for the carbon footprint
Oh,
because it's twice higher than pork and lamb, 4 times higher than dairy product and most plant product.
okay.
And so it's 10% of calorie. If you would replace that by a mix minutes per day, okay, and the carbon footprint by 1/3 by this small change. So think that pork could do in case of
In context.
one day lived in New Delhi.
One left, okay.
Okay.
Yeah.
Also, Beijing is 25 minutes lost per day with like a 50 microgram per cubic meter average. That we estimate. But if you are cooking indoor and you reach the 300 microgram per cubic meter, then you are rather at 60 minutes lost per day or something. But that's pretty cool. We can compare that. Yeah, that's.
Well, if it's bad enough, just eat whatever you want.
So if you have your chalet in Montana, you enter and there is this nice smoke odor. No, that's not nice. As a Swiss guy, I'm from Switzerland, and when we come to our Swiss chalet, I mean, traditionally you would smell the smoke, and so now we've put a particle filter, and we are the only chalet with 2 particle filter, the only particle filter of the whole municipality. Any other questions?
Yeah, I have one question. Just thinking about your models and maybe coming back to the hot dog model, which, pretty startling result, and I can see why there was a lot of interest in it. My question is how do you help people understand what the drivers of the model are? Because, you've taken something that has a lot of complex inputs and it rolls out with a very simple metric that then turns out to be very startling. So what is it how do you answer the question, well, what is it about the hot dog that is detrimental? And then, if it's based on sodium, for example, there's still a lot of debate in the public health community about how much sodium we should actually be consuming. So how do you help make sure the inputs that you have are good, and how do you help people understand how confident we can be in the outputs that come from these models?
Yeah, two things. First, we have stick really to the GPD, to the global burden of disease, in the sense, I think it's a good representation of the present level of science. They only select when they have a certain level of confidence. For a risk factor. They are pretty restrictive, in fact, in the way they do that. I think it will evolve, the first question. As science is coming, we can incorporate that. The minutes of life are interesting. I was speaking with a nutritionist who was saying, I've tried to speak to kids for ages. I've tried your metric, it works. I mean, the 8 years old pixie touch. Okay. And in a sense, it's an interesting metric in the sense that you, instead of combining and you have I mean, take a label and you look at the saturated fat, you look at, but for example, saturated fat, at one point we have in fact a sensitivity with saturated fat. There's so much debate there, and it's not in the global burnout disease that we've decided not to, I mean, create a risk factor for that, because, I mean, I'm just looking at a project with Nutella, in fact, to look at the individual fatty acids, to look at if there is a link with individual fatty acid and mortality. It might be hard to get, but it will be interesting to look at things like that. Yes.
On the risk that you have for each of the individual items, it's all based on a linear extrapolation. So, but for most of the things we know in biology, they're actually a threshold. So if you really in a major way, overestimating risk.
You mean for the food?
For the food.
Yeah, for the food.
For any of the thing, I mean, particles, for environment, for almost anything chemical, there's usually a threshold involved.
Yeah, so this would be, if you like, the threshold for processed meat is 2 grams per day. The present consumption is around probably 15 or so grams per day, the average consumption in the US. So that's why I was saying that for most people, in fact, they are in the active range where they are above threshold.
Yeah, but when you're making the calculation,
yes,
you're assuming that it's whatever it is, it's extrapolating from 0 to 100, whatever it is. And then you also don't take into account essential ingredients. So for sodium, do you factor into the fact that you have to have a couple grams of sodium to survive?
Yes. So for the sodium, for example, the first, I don't remember how many grams, will not be counted. So you count those over a certain quantity in that case. So you have a minimum. The global burnout disease has called TMLR, theoretical minimum or maximum limit, above which you have no effect for the beneficial, and below which you have no effect for the detrimental. Well, what we don't take into account is the interaction. I mean, and we know the epi, you correct for some factor, but not also. Is it really the vegetable who are good, or is it because people who eat more vegetable eat less of something detrimental? And there we could double count. That's where I think machine learning and taking population and the measure would be really cool. But the problem with food is the measurements of the consumption is not that easy. Yeah. It's a challenge.
Okay, any other questions here? Any questions from the folks online at all? Let's see, nothing popping up, so. Okay, great. Thank you for a fascinating presentation. I really appreciate it. So we're running well ahead of schedule right now. So we're gonna stick to the current schedule. So at this point, we are on for a break. We ready? Okay, great. Well, good afternoon again, everyone. So we have 2 more sessions this afternoon, and it's first my pleasure to introduce Jeff Keithline, who is from the law firm Keller Heckman based in Washington, D.C., where he specializes in the area of food safety and drug law. As I was looking through Jeff's resume, I'm going to summarize it by saying he has a very broad portfolio in a broad range of food-related, drug-related, and regulatory-related activities. And he helps companies meet regulatory requirements at the U.S. federal level, including USDA, FDA, EPA, big portfolio there. And then it moves on to state and regulatory work, and then he works internationally. And it's Canada, Mexico, EU, Pacific Rim, North America, South America. So it's a really broad portfolio, and it's really impressive that he manages it so well. And then one other interesting thing about Jeff is that prior to law school, he was a commissioned officer in the U.S. Navy, where he served aboard the nuclear submarine USS West Virginia. So we thank you for your service, and we thank you for being here this afternoon. Yes, that would be a good thing. So let me just shut it off.
The nuclear sub thing is always a great icebreaker. I can tell you all kinds of submarine stories at a break sometime. It's happy to do it. Well, I'm not sure if you were an early adopter of signing up for the registration for the program, you would have seen that my colleague Devin Hill was actually scheduled to speak, but unfortunately, Devin's actually been active, I think, with this group for many years, but unfortunately, he was triple or quadruple booked, so he asked me to come to Michigan. I'm always happy to come to Western Michigan. My wife grew up in Grand Rapids, and so it's nice to come back to some of the old stomping grounds. I'm a little bit in awe here because I think I'm surrounded by a lot of Ph.D. chemists or Ph.D. toxicologists. And I'm just a mere lawyer, so it's a little bit different here. We do have in our staff a lot of staff chemists, and so we talk a lot of science in our firm, but I am not a chemist. You hear me? All right, so let's jump into it. My talk is going to be talking about the regulation of food contact materials, but I wanted to talk a little bit about, the format's a little off, but we'll figure it out. So talk a little bit about how food contact materials are regulated in the US. It's going to be a little bit of overlap from the couple of speakers earlier this morning, so I'll kind of breeze through it a little bit. FDA clears food contact materials as food additives. I mean, that's where they get their regulatory authority. So if it doesn't meet the definition of a food additive, it's not required to be regulated by FDA. So the definition of a food additive is a substance. This is a pared down version because it actually goes on for paragraphs. But a substance, the intended use of which results in or may reasonably be expected to result in that substance becoming a component of food under its intended conditions of use, unless that substance is not generally recognized as safe. And so as lawyers, we also try to find the loopholes or the exemptions there. So built into that, there are several different exemptions from the definition of a food additive. FDA clears food additives under their authority to regulate food additives, to clear substances under the food additive regulation process. There also is a threshold of regulation process, which if you could demonstrate that your use resulted in a very low exposure, FDA would issue you a letter said you're exempt from regulation. That's largely been supplanted by this new food contact notification program, but it still exists today, and you can still get a TOR if you submit paperwork. Sometimes FDA there may be good reasons for doing that. One different aspect of the TOR program is it's not proprietary, and so if you don't have a lot of detail about the manufacturer of a substance, you may choose to do a TOR rather than the food contact notification program. And then again, there's exemptions from regulations, and that's what we spend a lot of our day doing, is telling people they're exempt from having to go to FDA to get a food additive clearance for their materials. As I think we heard earlier, the food contact notification program is pretty new. It was first adopted in 1997 in FDAMA, and first was implemented in the year 2000, with the first notifications becoming effective, I think, in April or May of that year. And now we're up over 1,000 FCNs that have been adopted. If you looked earlier in the version of my title, it included NIOS, and NIOS is not really a term that's well defined under FDA regulations. It stands for non-intentionally added substances, and NIOS are all the rage in Europe. Everyone in Europe is talking about NIOS and how to deal with those. And it really comes to us from what was known as a plastics implementing measure, which resulted in the development of the EU plastics regulation, which was first adopted in 2011. And it's defined as an impurity or a reaction intermediate product formed during the manufacture of that product. NIOS are not required to be listed in Europe, but they must be risk assessed under Article 19 of the plastics regulation. And so when people talk about NIOS, this is really what they're talking about. They're talking about impurities, which is really the subject of my talk. So basically, in the US, we don't talk about NIOS as much, but they're the same thing. They're dealing with impurities. The FDA regulation for food contact materials, the good manufacturing practice regulation, is under 174.5. And that regulation is really the overarching regulation. It's very much like in Europe, the framework regulation, which talks about what you can and can't do with food contact materials. It essentially says you must use the minimum amount necessary to accomplish a technical effect, which that's actually one that people don't really talk about much, but that's one of the aspects of that regulation. It also says that the substances must be of a suitable purity, a purity suitable for its intended use. And then it also mentions that the existence of a food contact regulation or even a food contact notification, which doesn't really say it in the regulation, but it's implied, doesn't alleviate you to look at other aspects of safety and suitable purity. And they mention a couple examples in that regulation. One, you can't impart adverse taste or odor to food, which is something, certainly that's very common in Europe for people to do that testing. In the US, that testing is often done by the consumer brands folks, not so much the packaging folks. But in Europe, occasionally that work is done at the raw material supplier level. It also says you can't render the food as adulterated under Section 402 of the Act. So because my presentation is talking about the food contact notification program, I thought I'd go a little bit into the data requirements for the FCN program. FDA does, as we heard earlier today, has a chemistry guidance that and they also have a toxicity guidance and admin guidance, but primarily I'll talk about the chemistry guidance, which provides details about how, what information needs to be included in a notification to FDA to assess not just the food contact substance, but also the impurities. This is an excerpt from the chemistry regulation or the chemistry guidance that suggests that not just the information on the identity of the FCS, but also any impurities as well as its composition by weight that exist in the material. And they give several examples. They say residual starting materials, catalysts, adjuvants, production aids, byproducts, breakdown products are all identified. And they also indicate that solvents need to be assessed as well as residual monomers. But for polymer adjuvants, the agency indicates that they're likely not going to be an issue, and you don't have to provide information on those adjuvants unless you have significant toxicity concerns. And of those, they mentioned carcinogenic impurities as well as degradable polymers and reactive substances. And it's our experience at Keller and Heckman, many people are now looking at these degradable polymers as something that they're developing for packaging materials. Your either bio-based polymers or degradable polymers in some way, compostable polymers. And we're finding that FDA is asking more and more questions about what happens to the fate of those during the actual packaging of the food as well as its end of life. And so you have to consider whether that product is going to degrade when it's in contact with food. And they're asking lots of questions about that. So this is kind of an eye chart. You really can't see much of this, but one of the required elements of a food contact notification is FDA's Form 3480. FDA ostensibly has the authority to reject a notification if you don't include this form with your notification. And it's almost as a double-edged sword. We, as part of the rulemaking that led to the FCN program, Keller and Heckman suggested to FDA that they use a form because we thought it would be a really good way of kind of summarizing information and just essentially be a form that you could provide and get that and be a very abbreviated process. Well, now it's become kind of almost a burden to have to do this, and now it's been a bit more simplified now. But this is an excerpt from the form. It indicates that you need to provide information. I'm not sure if folks can see it here, but you need to provide information on the chemical name, the CAS number, and then the typical and maximum residues of that impurity in the food contact substance. And then you need to go on and provide additional information. So if there's some concern about the stability of the polymer, if it's going to degrade, you need to provide information about that. And that's particularly important when you're looking at an antioxidant. So if you have an antioxidant that degrades, or as it functions, it actually oxidizes, you need to consider that oxidation byproduct in addition to just the food contact notification, food contact substance. So you have to provide information, that's the degradants. And then finally, you need to provide information about migration of that substance. And then ultimately, you need to provide information about the toxicity of all the substances that migrate to food. When assessing impurities, I think I've mentioned some of these already, residual monomers, catalysts and their residues. So catalysts typically are there, but initiators will often break down and form byproducts. And you need to consider those when you're assessing these impurities in food. Just, I guess, one more aspect of the food contact notification process to differentiate it from the food additive petitioning process is that it's proprietary. And so when I provide information to FDA about my new food contact substance, I need to provide specific details about how I make that product. And because in the old petition days, I was petitioning not just for myself, but anyone who else could make that substance. And so I'm really providing information about the end product. Now what I have to provide is information about how I produce a product, and I really need to assess any of the impurities that are result from my manufacturing process. And so that's a bit different process than the way we used to do it. When we're evaluating the identity, as you may recall, I mentioned you have to identify the impurities as well as provide information about the concentration. And so I'm not a scientist, but we talk about this in the office, and we submit lots of FCNs. But so if you have known impurities, like say a residual monomer or an antioxidant byproduct, you need to have analytical standards of those, and then look for those substances in the food contact substance and provide information. And that's really straightforward. You have an analytical standard, you develop a calibration curve, you find out what their retention time is, and you look for those substances. The harder ones are the unknowns, because if I would provide FDA information that I have an additive that's 98% pure, the first question I get back from FDA is, well, what's that other 2%? What could potentially be there? And they want you to look at the 100% concentration or the 100% composition of that product. And so that becomes a challenge when you're looking at for unknown impurities. And so typically we have people look for just using a survey method, comparing against library matches. GC mass spec, HPLC, LC mass spec, I think is another one that's commonly used. So those are comparing against library matches to determine if they can find those. The challenges with that, from my perspective, is you're comparing. You don't have a known level, so the quantification of that sometimes is difficult because you don't know exactly how it's performing. And typically it's validated, but it's validated against an internal standard. And so if you're using an internal standard to validate that method, you're not going to get quantification right down to the very low levels. So that can be a challenge. FDA requires in migration studies that you conduct validation. That's typically spiking recovery validation. So you spike known concentrations of the material in and demonstrate that you can adequately recover that. And that's what exists in FDA's regulations for migration testing. But we've also found that FDA now expects folks to do that same spiking and recovery type validation when you're doing residuals analysis to make sure that you're adequately quantifying the level of those residues in your materials. Also, when you're doing an extraction study, they want to make sure that you're exhaustively extracting that material as well. So you have to demonstrate you're doing that. And once you have the quantity of the impurity in your food contact substance, then you need to conduct migration calculations or studies to determine whether those substances are getting into food. We heard a little bit about that, how to do that today. We talked a little bit about actual migration testing when Dr. Duncan mentioned some migration testing, and others have done that as well. And then modeling, a few people have talked about diffusion modeling. The easiest and most straightforward is just assume 100% gets into food. And sometimes that works with impurities, but not always. And then evaluate the toxicity. So I just threw this up because this is kind of what we're sometimes dealing with. This used to be called the forest of peaks, and you have a substance and it looks like it's pure, but no, you have all these things you need to evaluate. I think there are 29 different peaks here identified, and that becomes a challenge. And this, a forest of peaks type of a chromatogram like this, I went to a talk back in, I think, 2005 in Europe, and they were talking about how to deal with this. And someone threw up a chromatogram that was even noisier than this and said, how are we going to deal with all these things? Well, one way is to draw a line across above the baseline and say, here's our exposure line below which we don't have to worry about the toxicity. And so we only have to identify the ones that are above that line. And that assumes that your extinction coefficient, that your identity of those substances and quantification is adequate. But that's at least one way to deal with those substances. So I think we talked a lot about exposure, and FDA evaluates things on an exposure basis, but they do it somewhat differently than different parts of the world. And I thought it might be helpful just to talk a little bit about how that's done by FDA. So they they use, as I think several people have talked about, food simulating solvents to determine migration level in food. And they also use information about packaging factors. And we haven't talked a lot about packaging factors today, but FDA has done surveys about how packaging is used, and their chemistry guidance includes information about that. And so they assume that 21% of, 16% of our diet is packaged in PET. And they've then subdivided that across different types of food. So they say that most PET is used to package carbonated soft drinks. And so that's an acidic food. So 97% of that food is acidic, where only 1% is fatty, 1% is aqueous, and 1% is alcoholic. And so they have used those factors to be able to take your information about how your individual substance migrates to different types of food simulating solvents to calculate what the exposure is. And they've developed that for a number of different types of polymer systems. For polyolefins, it's about 30% fatty foods and 60% or 33 and 66% fatty to aqueous food types. Typically, people don't do all food types, all four food simulants. They typically do a 10% ethanol to cover both acidic foods and aqueous foods, and then they do a fatty food simulant, oftentimes a alcohol in water, 95 or 50% ethanol solution to cover fatty foods and alcoholic foods. If you can actually get a method to work in a fatty food simulant, in a true fat like olive oil or a similar type of food oil, then that's accepted. But oftentimes people don't want to shoot oils onto their chromatograph, their LC mass spec or GC mass spec. So you take that information, once you have the migration level in the various food simulating solvents, you multiply them by these food type distribution factors, and that gets you a weight average migration limit. The weight average migration limit is then multiplied by a consumption factor. The consumption factor, again, is those packaging factors that indicate how much individual polymer systems are contacting or used to package our diet. If you actually sum up all of the packaging or the consumption factors in FDA's chemistry guidance, you get about 160 or 180% of the diet, which is kind of, that just reflects history. What happens is FDA almost always, when they figure out something, a new polymer is taking hold and it's being used more, they'll add to those numbers, but they typically don't subtract from them. They are undergoing a big effort right now to reconsider what the packaging factors are. And for some polymers, it's going to drop significantly. I think polystyrene is going to drop significantly. Paper is going to drop significantly. So paper is currently in the guidance document 30% of our diet. So 20% of our diet's packaged in polymer coated paper, 10% in uncoated paper. And I think that's actually going to drop significantly when they revise those factors. But I always use the adage when I say the consumption factors added to more 100%. I said that's why there's an obesity factor in the United States, because we're eating more than we're supposed to. So you take that information, that gets you the dietary concentration. Typically at Keller and Heckman, we report that in a concentration in the diet. So we often talk about it in a parts per billion in the diet, but really it's expressed as a dietary intake. So you multiply that by 3 kilograms of food per day, which is the FDA assumption, and that gets you an intake. And then you can divide it by a typical FDA assumption is 60 kilogram body weight per day, and that gets you grams or milligrams per kilogram body weight. Just a side note, if you're looking at infant exposure, infant applications, FDA has updated their guidance document to indicate that when you're assessing packaging for infant formula or breast milk, you need to do a special assessment. And they really assess that using much lower thresholds. And the concern there is that you're exposing a subpopulation that is somewhat sensitive. They eat a higher amount of food relative to their body weight, and likely 100% of their diet is packaged in a certain type, in a particular package. And so all of those together really kind of push down the levels of exposure that you need to consider. So I think we talked about this before. You need to look at all the data. When you're submitting a food contact notification, you need to assess the toxicity of all, not just the food contact substance, but also the impurities. And I think one of the FDA speakers today also mentioned you need to look at this as a cumulative exposure. So you need to look at not just your exposure, but how that's added to all the other uses. We talked about that earlier. So threshold toxicants like non-genotoxic carcinogens, we look at that using an acceptable daily intake. Basically, you take the no effect level or no adverse effect level and apply safety factors depending on the length of the study. And for for non-threshold carcinogens and mutagens, FDA's got a different risk assessment process. For cancer, they essentially use a straight line approach from high dose animals all just straight down and assume a 1 in 1 million risk level is what's acceptable. But typically, if you're submitting information for a new food contact substance, FDA really doesn't want you to consume more than a 10 to the minus 7 risk because they want to allow for other uses. And if it's a high use material, FDA may even draw you down to a 10 to the minus 8 risk to make sure that that there's some additional room for, or just to cover to make sure it's a de minimis increase in exposure. FDA also looks at genotoxic substances. I mentioned, if we have a substance that's genotoxic, FDA's default is 50 parts per trillion in the diet. I mentioned before the NIOS assessments in Europe. There's been a new recent change to the plastics regulation, the 15th Amendment, which suggests that if you have information that, or if you cannot rule out that a substance is non-genotoxic. So if you have, not that you have evidence that it's genotoxic, you have to rule out that it's not genotoxic, and you don't have evidence that the substance is not migrating to food at more than 0.15 parts per billion in the food, then you need to actually disclose the identity and concentration of that substance. And so that is becoming a big deal for the industry in Europe, and it's requiring a lot of people to go back and look at the potential migration and exposure of impurities in food. So we talk, this is just briefly, I think we talked about this from FDA speakers. They have an increasing risk tiered approach to assessing toxicity. If your dietary concentration for your use is less than 0.5 parts per billion or 0.025 milligrams per kilogram body weight per day, then you don't have to do any testing. You don't have to do any genotoxicity testing, and you also don't have to accumulate that exposure. They consider that to be a de minimis increase. But at the higher levels, you have to do more and more toxicity testing. So and even for an impurity, if your impurity is greater than, say, 0.5 parts per billion, you have to have evidence about its genotoxicity. So FDA typically wants to see the Ames test and then another in vitro study, typically chromab or mouse lymphoma. Sometimes people have provided the mouse micronucleus in vitro as an alternative. If you are higher levels, you have to provide subchronic feeding studies as well as all of the tiers, all the information below, as well as the in vivo mouse micronucleus is typically provided. And if you're above a part per million in the diet, typically FDA will not accept a food contact notification for that level because they believe that it takes a chronic feeding study to support a dietary exposure at that level. And what you're going to have to do is, FDA is going to take more than its 128-day statutory review period to review that carcinogenicity bioassay. And so they ask that if you're going to do that, you come in and speak to them first before you actually submit that data to FDA. So this is unreadable on my screen. Maybe it's better, it's readable here. So this is just an example of how you do the calculation. So if you had a current dietary exposure of 40 parts per billion, and you had migration data indicating relatively low migration to 10% ethanol, but high migration to 95% ethanol in HDP or in LLDPE, then you take those factors, the various food type distribution factors and the consumption factor, and you calculate the dietary concentration for your use, which here was 60 parts per billion. You add that to the existing exposure, that takes you 100 parts per billion, and that's in the subchronic feeding study area. So you have to have a sufficient amount of data. I would be remiss not to talk about what happens if you make a manufacturing change. So I said before that FCNs are proprietary. They deal with the manufacturing process that you submit to FDA. If you make a change to that process, your FDA it includes information in their admin guidance that says you need to make sure that you assess the potential impact on the levels of impurities and the identity of impurities. And so if I switch from one catalyst system to a different catalyst system, I need to assess lots of things. I need to assess, am I generating different impurities? Am I as efficient in the polymerization process? Are my residual monomer levels the same? Is the molecular weight, the low molecular weight oligomer content of that polymer the same? So I need to do all those levels. FDA also, in its final letter to the notifiers, they're not obligated to provide a final letter, but they do. And in that, they also remind you, hey, if you change the manufacturing process, you need to let us know, you need to assess the impurities and let us know about those changes. But they also say in their admin guidance that changes that are considered within good manufacturing practice are acceptable. So typically our response is, if you make a change to an impurity level and you don't go into a new toxicity tier or you don't increase the exposure to impurities, then you're not obligated to go to FDA and let them know about that change. That would be considered within the scope of the existing notification. But if you make a change that would increase or change the identity of impurities, oftentimes you need to go to FDA. What FDA is likely going to say to you is that that's very good. We're going to consider this a new FCN. We're going to change your FCN and give you a new FCN number. So I'm not sure how long it took me to talk, but that's the content that I had to go through. I'm happy to take any questions if there are any, and happy to chat with folks afterwards.
Thank you very much.
Thank you.
So I have one online question to start off with. It's a little technical, but we'll go through it here. It says, could you please tell us whether GMP requirements for food in 21 CFR Part 110 are applicable to the manufacturing of food packaging? And then there's a statement that follows. Yeah. I understand suitable purity in 174.5 is required, but I'm wondering if GMP in Part 110 is required for food packaging and manufacturing sites.
It's a great question. So just FYI, the good manufacturing practice requirements in Part 110 have been now moved into Part 117. So if you're looking at the old 110, you'll see them now in Part 117, which FDA has done as part of the updates to the requirements recently. Typically, we tell people they're not required to follow those food GMPs for the development of food contact materials. If you look in that section, there are sections that deal with food contact surfaces. That's really directed to the food producers. So if you're producing food on a conveyor belt, you need to make sure that food contact surface is sanitized between batches of food to make sure you're clearing. But it doesn't necessarily follow that you have to do everything in Part 117 for the production of food contact materials. We also say, however, that it's a really good place to look because they do tell you about record keeping, about training, about making sure you assess different things. And so although it's not necessarily applicable to food contact materials, it's a good place to start when you're trying to develop a GMP plan. Yeah.
Okay, that's all the online questions I have over here.
I do have a question. Do I need to use the mic?
No, we're going to use
okay.
We're on the overhead.
Okay. I'm just wondering if you have any idea, that slide that you showed of the sort of the tiered data requirements, how frequently each of those are triggered in general?
Oh, so how frequently these are triggered. So almost always we're trying to get in one of the lower two tiers because those are the lower cost requirements. Right. If I'm making a significant, and typically polymers are down there. So FDA has published a paper that says if you have data on oligomers and your dietary exposure isn't at less than 50 parts per billion in the diet, you can rely on the toxicity of the constituent monomers rather than looking at generating toxicity data on the oligomers. But if you have a plastic that's greater than 50, FDA might suggest that you no longer can rely on that. So you may have to extract significant amounts of that, develop quantities of the low molecular weight fraction and actually feed oligomers to rats. And that's going to be hard. But so we try to, for polymers, to try to keep the low molecular weight fraction in those lower two tiers. If you're looking, there are a lot of antioxidants that are here or even here today because they're very highly used. Argonox 1010, for example, which is a very common antioxidant used in the production of most plastics, they have a chronic feeding study in that support. But a lot of other ones that I've seen have greater than 50 parts per billion in the diet. And so again, this is probably a $500,000 effort to be able to develop that data. And but most people today are finding ways to keep below that. We're not doing a lot of rat studies today or mice. So typically, if you're on the lower end of one of these tiers, you might be able to go into FDA and argue, hey, I'm just right on the edge. I'm only going to do one of the studies because they'll require two subchronic feeding studies at that greater than 50 ppb. But they've kind of had an informal approach. If you're less than 200, you probably can get away with just a rat study instead of the rat and mice, a rat and dog study, because it's a rodent and non-rodent.
Today is rat and dog.
Rat and dog, yeah. So that's what this is. So most people are not doing dog studies anymore. But the other aspect of this is, because it's a proprietary system, I can tell FDA, although the consumption factor that I might typically use is say 5% or 20%, depending on the packaging I'm selling, I might say, I'm only going to sell X percent of this or X pounds of this into this market. And so then I can maybe move that down to avoid that animal testing, because they typically assume that you're going to consume the entire market if you're making a new material, which is unrealistic. And so if you're making a new polyester or something, you may not be consuming lots of the market. I can't give you a percentage, but typically it's the in vitro studies are what supports probably, I guess, 95% of the FCNs that are submitted today. And if we're higher than that, we're probably relying on existing data that exists that already been provided for other purposes, maybe REACH or something like that. So not for this purpose.
Thanks.
Olivier? One question on data availability. At one point, I think FDA was providing the ADI in a list, and I think it has disappeared.
That's correct. So they did provide not just an ADI, they also provided the cumulative estimated daily intake on their website. It's off their website. They don't do that anymore because they're not updating it. The reason they took the ADIs off their website is because they feel that was backing them into a corner. They wanted to say, we want to take an opportunity to relook at those data to make sure that we are continuing to support those. They don't want to lull notifiers into believing that FDA established an ADI for a substance in the 70s, but they're going to still support that today. So that's why they pulled it down. If you have a question, you can go to FDA and ask for that, and you can go to the Internet Archives and find where that document still exists on their website.
I tried to look, but I couldn't find it.
I can see if I can find you a copy of it and send it to you. Yeah. The CEDI also was incomplete, so they didn't provide complete information about the cumulative exposure. You can go in and submit what's called a pre-notification consultation, which is asking FDA for information and ask them, hey, we're interested in submitting a notification for substance X. Can you tell us what you have on the toxicity of that substance, if you have an established ADI, and what the existing cumulative exposure is? And they will tell you. It often takes 4 or 5 months for them to respond to that, but they will let you know.
Okay.
All right.
Anything else? Okay. Again, thank you very much.
I think that's yours, right?
We're up there next. Okay, good. All right. It's now my pleasure to introduce Karen Hagerman, who is a senior manager at an organization called Green Blue, which is an environmental nonprofit dedicated to the use of sustainable materials in society, and it's affiliated with the parent organization Sustainable Package Coalition, or SPC. What SPC does is it brings together members of the packaging value chain to address critical issues within the packaging sustainability area. Karen holds a bachelor's degree in aerospace engineering from the U.S. Naval Academy and a master's degree in sustainable engineering from Villanova University. Prior to joining Green Blue, Karen worked in operations and program management as a Sikorsky CH-53 Super Stallion helicopter pilot in the United States Marine Corps. So we too should thank her for her service to the U.S. and to the nation. And we also thank you for being here today. So. Much.
All right.
And you've got it all. Okay.
Yes.
Thank you.
Great. Thank you so much for that introduction, Leon. Let me get this clipped in. Thanks to both Leon and Norb for having me today, to MSU and the center. It's really a privilege to be here. And for me, it's really exciting to have gone at the end of the day after hearing all of this exciting science and research that's going on and seeing how I can learn from it and how I can bring that back to the members of the Sustainable Packaging Coalition and think about this research and how we can bridge that gap into action that results in safer and more circular materials in society. And so again, Leon gave my introduction, all that to really say that this area of material science and safer chemistry is not my background. I feel very in awe of the amount of research and detail that you all have gone into and that I've heard of today. I think it's fascinating. I do not have much experience with toxicology or with material science, but what I do is I lead a stakeholder group that's focused on bringing industry and stakeholders together from throughout the packaging value chain to look at how we can accelerate the adoption of more safe and circular materials. And so that's what I'll be talking about today. So just for a little bit of background, as Leon mentioned, I work for Green Blue, which is the parent nonprofit of the Sustainable Packaging Coalition. The Sustainable Packaging Coalition, we are up to about 560 members from throughout the packaging value chain. So that includes multinational consumer goods companies to smaller brands to packaging converters, raw material suppliers, retailers. We also have academic members and government members. So the point is really to be that nexus of where these stakeholders can come together to engage on these different issues in sustainable packaging. Under Green Blue, we also have a couple other initiatives that you might be familiar with. One is the How to Recycle label, which is a way to communicate to consumers how products can be recycled and awarding specific labels to those products. We also have the Clean Ingredients Initiative, which is a database of verified chemicals that are safe for use in cleaning products. And so a formulator can go into that database and develop a formula for a cleaning product that then can be used to achieve the EPA Safer Choice label. And then one of our newest initiatives is the Recycled Material Standard, which is a chain of custody certification for recycled content to be traced throughout the system and then labeled so that in its future use in new products, it can be verified that that is in fact recycled material and where it came from. So the focus of what I'll be talking on today is the role of material health in packaging circularity. And I think it's been touched on a lot throughout the day in terms of life cycle impacts and in terms of exposure to human health and the environment. But I'll touch on that a little more and then share with you the work of the stakeholder group that I mentioned, which is our Safe and Circular Materials Collaborative. And then from there, I'll go into a little bit about ChemForward, who is our partner in this collaborative. And they have developed a pilot project that they're working on in conjunction with the stakeholder group. And I'll share a little bit about what they're doing and what the purpose of that is and what they're currently working on. So as we mentioned, there are so many different considerations when it comes to how the chemistry and the materials within a package impact the world, whether that's the humans, whether that's animals or the environment or all of the above. At Green Blue, we really look at packaging and materials overall in terms of its entire life cycle. And so this little diagram shows really all of the things we're thinking of in terms of sustainability. And so you can see we're looking at sourcing, manufacturing, distribution, use, recovery, also transport, and really overall design and how companies and really the entire industry can think through packaging materials that take into account impacts throughout the life cycle. So we view sustainable packaging as packaging that is sourced responsibly, that is optimized for efficiency, has recovered effectively at its end of life, and then is also safe for human health and the environment throughout its life. So those kind of 4 pillars are a big part of what we look at. We get a lot of questions from members or from non-members asking, what is the most sustainable packaging? What should I use? And I'm sure you guys can guess the answer to that is it depends. It depends on the use. As we mentioned with some of the migration, it depends on what the substance is. And so really looking at what are we hiring what is the job that we're hiring that packaging to do? What is its purpose? And then really looking at its entire life cycle to understand how is it impacting the environment? How is it interacting with humans? And so there's so much going into it. And that's a really big component. So when we're talking about sustainability, it's not just that it can be recycled, or it's not just that we sourced from a responsible forest. It's really what is the impact throughout the entire life cycle. So when it specifically comes to material health and packaging design, there's a couple aspects that we think of when we're asked, what does it mean to design with material health in mind? And here, if it's not familiar language, we use material health basically to represent green chemistry or the chemistry that's included in the packaging and how that impacts and interacts with, again, humans and the environment. And so the first aspect of that is to choose inherently safer materials. And so starting out with eliminating the materials of concern, avoiding chemical hazards when you're developing a packaging, developing the material that will be used in packaging. And for that, starting with those restricted and those chemicals that are known hazards and on regulatory lists, that is a great place to start. But there is a lot that can be done beyond that to ensure safety. Sometimes, as we know, and I think we've talked about today, a lot of the science is well ahead of where regulation is. And so there's a base level where complying with regulation will get us to a certain level of safety, but we really need to go beyond that. And that requires thinking about these chemicals, understanding the hazards associated with what you're using, and doing that early enough in the design that it's really foundational to what you're putting into your packaging material and what you're putting out on the market. But as we know, there's a challenge there because choosing safer alternatives is easier said than done in a lot of situations. A lot of that information is not readily available. And for companies that really want to move into safer packaging, they have to go above and beyond. They have to go out of their way and do extra work to make sure that those materials are safe. And so that kind of ties into the next aspect, which is building the supply chain transparency. So it's difficult to assess the safety of a package without understanding everything that's in the package. And when it comes to packaging, supply chains are very complex. There's often multiple tiers of suppliers, and getting the information is often behind. There's a lot of, or some level of reluctance to share the information because of proprietary information or trade secrets. But if a company or if the third-party assessor doesn't know exactly what's in the material, it's really challenging to assess that that material is safe. And then going into not just what are those chemicals that are in it, but when you look at packaging, there's so much more to it. And the more time I spend at the Sustainable Packaging Coalition, the more I realize that a package isn't just a simple plastic bag. There's coatings, there's adhesive, there's sealants, there's different closures, fixtures. There's so much more to a package than just the main substrate that it's made of. And so all of those things need to be considered when you're considering not just material health, but really all of overall sustainability, because those also have a big impact on recyclability, on compostability, and whatever the end of life is for it. So really thinking about the entire packaging system and not just the individual polymers or materials that are being used. And then considering the whole life cycle, as I mentioned, that's a really critical part of it. And I think when we're thinking of chemical hazards and packaging, we often think of the use phase, and we think of how it's, as we talked today, how that material is migrating into food during the use phase. But there are so many other considerations that we need to think of too, including any sort of chemical hazards associated with the manufacturing process, or the end of life, whether it's recycling or composting. And then I think, Norb, your example earlier about the transport, that's another consideration too. When you have different conditions that the packaging is being exposed to, how does that change it? And are we considering all of this? But that is a lot. And just hearing the little bits of science and research that's gone into the presentations today, it can seem overwhelming to really understand all of these different aspects. But regardless of how overwhelming it is, there is definitely an urgency and a need, especially for a lot of companies that are focused on having more sustainable options, to act and to do something to ensure that what they're putting on the market is safe. This ties into the idea of circularity. So I'm sure many of you are familiar, but the idea of the circular economy, where materials maintain within the system. So materials are either recycled back into new products, or materials are composted and then turned into biomass that can then be used to develop new bioproducts, or just fertilize land or turn back into soil. There's a strong push for companies to move towards circularity. There's global commitments on circularity. There's commitments on meeting certain recycling rates, recycled content requirements. But with this is kind of an emerging focus on, well, if we are circular in our packaging systems and our materials, what does that mean? What chemicals are also circulating around that system, and what should we be aware of? How do these chemicals that are in the materials, how do they impact the recycling process? What is their impact on the post-consumer recycled content that comes out of that process? And also, is there anything that happens, that occurs as the materials cycle around a few times around the system? Do they continue to degrade? There's a lot of unknowns there, but there's a lot more attention on that now as well. I'm sure many have read recent headlines and heard news about the issue of PFAS in paper food packaging. So it's used as a grease and moisture barrier in a lot of compostable products. And this is becoming more and more, there's a lot of attention and regulation concerning eliminating PFAS from these uses. And so a lot of companies are scrambling to find safer alternatives and to verify that those alternatives not only are a good enough alternative, but also have the same properties and perform like the PFAS, the perfluorinated substances were intended to perform. And so this leads a lot of companies to asking, what's the next PFAS? And this isn't just, there is an element of wanting to avoid, to mitigate risk and avoid the bad press that comes from this. But with a lot of companies, we're saying that there's just the desire to break this cycle, to get ahead from where we continue to every so often, every couple of years, have a new packaging chemical emerge as hazardous and having to move to something else. And so realizing that a lot of companies too want to go beyond legislation so that they're not caught off guard by the next thing that's regulated, but really looking at not being forced to not be hazardous, but really like moving towards a positive side, towards the safe and circular, and really looking and ensuring that whatever they're using, it is safe and it is circular. And this is kind of the last point too, is that when something's free of a substance, so if it's PFAS free or BPA free, that's great. We know that there's not that chemical hazard in it, but what is in that? And that's a big thing too, with a lot of companies wanting to avoid regrettable solutions. So wanting to know whatever I move to, ensuring that that is safe, and then that's not going to cause any sort of chemical hazard down the line. Part of that too ties into some of this research, and there's other papers too that I could list here, but really understanding a lot of what we talked about today with the impacts of food contact chemicals on human health. And this is a snapshot of a consensus statement from last year that really talked about how a lot of the global regulations are inadequate to actually assessing or preventing the risk to human health that's connected to a lot of packaging chemicals. And there's a lot of gaps in information that need to be filled. And this consensus statement also emphasized the relationship, which I think has not really been, as I mentioned, it hasn't solidified really in the past, the connection between chemical safety and the circular economy, that it's an essential component of transitioning to a circular economy. So the challenge here for a lot of our members, and which led to the convening of the stakeholder group, is really understanding that there's a need for chemicals to be, or for packaging ingredients to contain chemicals that are less toxic to humans, less polluting to the environment, and more suitable for circularity. So all of those things in one. And to do this, it requires putting safer, optimized feedstock inputs onto the market in the first place so that we can ensure that what's going through the system doesn't have these issues. And so that leads me to the Safe and Circular Materials Collaborative. And so an overview just quickly of what a collaborative is. So as I mentioned, the SBC has a large membership. We have members from all different types of materials and industries in terms of who they serve, whether it's food packaging or automotive or apparel. So people have a lot of different interests within the Sustainable Packaging Coalition. So we convene smaller groups to focus in on specific subsets of information or of subsets of topics that are important to them, relevant to them. And as I mentioned before, the way we view sustainable packaging has these components of being sourced responsibly, recovered effectively, optimized, and then safe for human health and the environment. And this is a breakdown of our current collaboratives, or our current stakeholder groups. And you can see there's heavy emphasis on the effective recovery. And that's because it's a really big challenge that a lot of companies are trying to figure out. They have these recycled content commitments, they have these recycling rate commitments, there's global recycling rates or global commitments in terms of using recycled materials. And there's a lot of challenges with recovery. So there's a lot of focus there, but what we realize too is that there needs to be more focus on the material health aspect. And I would argue that all of these collaboratives, all of these topics touch material health and they're all incorporated into it. And that's what we try to do. But one thing we did recognize is the need to move into this space and to really get this on the top of our member companies' minds and in their discussions when it comes to circularity and sustainability. So Stacey Glass and Lauren Hine, who are of Chem Forward, they're our partner organization in this, and they're an independent science-based nonprofit. Their specialty is really providing consistent and reliable data to companies through a centralized, globally harmonized repository. And so Stacey came to us asking, we have this infrastructure where we are providing chemical hazard data in building this transparency. We would like to move into the packaging space, but the SBC, you have a membership. And so what we wanted to do was join forces and develop the working group that in one sense, or one part of the group is discussing these challenges, identifying what needs to be done. And then the concurrent part is the pilot project, which I'll explain, but they're both kind of feeding into each other. So we'll talk about the collaborative, which is a stakeholder group first, and just a little bit about our goals. So the collaborative is planned to be a year long, and that's not saying that we're going to solve anything within a year or figure out all the problems. But the idea was that there's so much to understand in this space. There's so many different things that we need to take into consideration. So the first year of this collaborative is to really just take a comprehensive approach of what is the situation we're dealing with, how can we start to make decisions at a company level or take action at a larger industry level to ensure that we're moving towards safe and circular packaging. And the focus here really is on two big streams in terms of recovery. So looking at compostable packaging for its ability to contribute quality feedstock material back into the biological cycle, and then also looking at recyclable packaging for its ability to again, contribute high quality material into the post-consumer recycled content for new products. And so the goals were pretty broad in terms of what we're looking to achieve, because again, really it's exploring the space and understanding what the challenges are and what needs to be done. And so the first goal is really focusing in on the areas of highest concern and the materials or chemicals that have the potential to contribute the most towards circularity. The two middle goals here are more tied to the pilot project, so demonstrating the availability of verified safer alternatives, and then also taking those safer alternatives, putting them into a database or repository of verified information that then can be used by companies to figure out how to build packaging that is verified safe or safer. And then the last goal being to establish a common agenda to move the entire industry forward and to accelerate the adoption away from chemical hazards and towards safer alternatives. So these are our member companies and organizations. We have, as you can see, we have packaging converters, material suppliers, brands, retailers. We also have some other nonprofit partners like the Food Packaging Forum and NSF International as well. And so all of these organizations have an interest in or some connection with this space and working towards finding solutions. and this is just a brief overview of what the group has been working on. So we will say we're halfway through. So at this point, I don't have fantastic learnings to report out, any earth-shattering revelations. We are still kind of working through what these challenges are and what we could do as a group. But as you can see, the pilot project is occurring concurrently as well. So one of the things we did as a group was look at where are the drivers for change. So if we have this system, the current system that's broken, that there are some chemical hazards, that there are concerns with the system, that we have materials, for example, like PFAS that are getting through. Then we have this ideal system where all materials are circular and safe for human health and the environment. What are the drivers for moving us from one system to another? And so this is just showing, it's the same circle, the circle graph I showed at the beginning, just compressed to make more space, but really looking at the entire life cycle and all the different players involved. What is driving change? And it's interesting because this input was from the members who operated different levels of the supply chain, telling us what they see as their pressure. So there's complying with regulation, as I mentioned, anticipating future regulation and getting ahead of it. But then there's also pressure throughout the system, pressure from brands that their suppliers are providing transparent and accurate data, that there's restricted substance lists that brands or retailers are using. There's pressure from NGOs on all aspects of the supply chain. And there's also consumer pressure too, to have safer materials in their home and for use with their family. So a lot of different drivers moving us towards this more ideal system. We also looked at where the barriers are to change. So we know that we want to change. We know we have these drivers moving us to change. So what is preventing this change from happening? And in the hopes is to, in identifying the barriers, start to address what are the solutions that we as an industry can put into place, or what are the solutions that we can call for, that we can recommend to the broader industry or to legislators, wherever, to help overcome these barriers. And you can see a lot of the barriers are kind of focused in that manufacturing part of the supply chain. So from the material manufacturers to the brand owners, that is a lot of our membership, but that's also where there's a lot of challenges in terms of transparency, in terms of availability of solutions that are safer, but still meet the performance requirements that are needed, costs in different aspects. So a lot of barriers, and kind of our next work that we're doing is to start connecting those barriers with potential solutions to see if there's ways that we can overcome them, or at least alleviate them and get to, again, moving more quickly towards that ideal system. These are some potential solutions that arose in some of our discussions. And so you can see the list here. Hopefully it's easy to read with a bright color, but one of the potential solutions is a repository of chemical hazard assessments of safer alternatives by CAS number, so actually connected to the chemical. Another would be a common list of chemicals of concern that the whole industry can align around to ensure that we're all moving away from these hazardous chemicals. We can agree on that. And then other options included establishing standard and third-party verified transparency disclosures, a certification for safe and circular products, or a repository for verified trade name building blocks. So similar to the first one, but instead of by CAS number, doing it by the actual trade name material. And so I put them in this order. This is actually kind of the feedback we got from the group as to what would be most immediately helpful to them. And so really what rose to the top was that repository of safer alternatives by CAS number, and then also that common list of chemicals of concern. And so what's interesting about these solutions is that they're not really an either or. They exist on a continuum of sorts. And so you can see that the common list of chemicals of concern and third-party verified disclosures, those fit into the free of type of claims. So those are where we're eliminating or moving away from those chemicals of concern and then verifying that we don't have those chemicals in our materials. The other options exist more in the safer claims space. So with the impact there being making sure that not only are we eliminating those chemicals of concern, but that we're moving to safer chemistry and improved feedstocks for circularity. And so this is something we're currently working on, is looking at these different options and understanding what needs to be done and what we could do in this space to contribute to some of these solutions, but also looking at if these solutions were to put in place, are there additional barriers that those cause? Are there issues with cost or with time or a number of different factors are the barriers to putting these solutions in place as well. And so that a little bit ties into the pilot project. So again, what ChemForward was doing is a project that basically develops that repository or database of safer alternatives. So the idea is or what they've developed is basically a database where a supplier can put their material into the database, have it assessed by third-party assessors and verified, and then it's in this database that then a company who wants to use that material can see all of the concerns that are associated with those chemicals or with those trade name items, depending on what it is. So I'll explain a little more, but a little bit of background on ChemForward and what they do. So what they really identified is that there's a gap in terms of the transparency and screening and the actual innovation of people or companies who are working to really innovate and ensure that we're putting out the safest packaging products possible. And the gap is really in this available, low cost, high quality chemical hazard assessment data. And again, the purpose of that database is to provide these chemical hazard assessments at a low cost and at a level of assurance so companies can trust that this is good data, that when they use these materials, they understand any of the hazards associated with them, but they also understand that they're safer than some of the alternatives. So in developing the pilot project and in the research that ChemForward did and the conversations we had with our members, a couple things rose to the top as priority functional areas that we wanted to prove first. So doing a proof of concept with a pilot of what are these high priority areas that if we can show there's safer alternatives here, we can show the viability of having this type of database that provides this level of information to the industry. And the things, there were basically three kind of functional areas that rose to the top. So for materials that are designed to compost, the challenge there that's on top of everyone's mind is really the PFAS-free performance additives to paper and pulp, specifically for food contact products. And then on the polymer side, really looking at the plastic additives that are used to improve that are applied to the plastics, and looking at having safer alternatives that result in better recycled content after it returns through the system. And then applying to both of those pathways, the composting and the recycling, having inks, adhesives, and coatings that can be applied to both situations or both kind of contexts and ensure that they don't inhibit any sort of end-of-life process or cause any hazards or concerns. So the first step was to identify those. The second step was to assess them. And so the way that this was done is for some of the materials that we asked our members to nominate suppliers, or if they were suppliers to nominate, to self-nominate their products or chemicals and to have them assessed. And so these assessments were done by qualified assessors to ensure that they're compliant, that they're looking at all the human and environmental toxicological information available, and then getting those assessments verified by a third-party assessor so that we can build trust again and make sure that people using the system know that the materials that are in there have gone through the process and with a high level of certainty that they are safer. And then the last step is taking all of that information and putting it into the database, like I said, to have it be accessible by companies so companies can look for the materials they need and the ones that they want to build the product and basically build that product. And I'll show you a little bit about how the database works. But the idea here is to really, the business case for it is that suppliers who are making these products that are safe can have their products verified and can put them in the system, and then manufacturers, brands, whoever it is that wants to use these products can go into the system. And there's that pull through on the other side. So we connect that gap of where there's data on safer materials and there's people who want to use safer materials, and they can have that information, the trusted information flow throughout. And so for the progress of the pilot project right now, the two primary functional areas that they're looking at are the paper and pulp fiber, which is really those PFAS-free alternatives. So particularly looking at barriers and coatings. And with this aspect of it, the path that ChemForward is taking is evaluating trade name coatings that have been provided by suppliers. And they've done they're currently doing 5 trade name materials that are in the process for assessment. And we are looking to share those results at our spring conference to be able to say that these producers of these trade name materials went through this system and they're verified as safer alternatives. On the other side, when it comes to the polymers, what the pilot project is looking at is these different additives, so plasticizers, antioxidants, and stabilizers. And instead of looking at the trade name materials, they're assessing the generic chemicals more by cast number and looking at what the safer alternatives are in there. There's 24 chemicals that have been prioritized, and NSF has been participating in helping out with this effort to be able to have all of this data into the system so that we can go in and look and kind of prove the concept that there's available safer alternatives. So what I want to show you is a couple screenshots from that plastics additive database to hopefully provide an illustration of what this actually looks like in practice. So again, it is a database. It has all of this information, this chemical hazard data in it. And what you can do is if you were developing a packaging material, and you were building that out and you knew you needed certain components of it, you can go in and search for those components. So you can search for the chemical that maybe you are using and you want to find an alternative for it, and it'll show you different alternatives. You can also screen, you can screen the formulation that you are using to see if there's any hazards in it, and then you can look for safer alternatives. And what you can also do is look by the function or polymer compatibility. So if you're developing a polyethylene product and you want to see what's compatible with that would be safer, then you have the ability to do that here. And then there's the way that ChemForward has displayed the different materials is using this harmonized hazard band. So you can see the color coding and the lettering, but they've also have equivalencies to the cradle to cradle certification, to US EPA skill list, to the green screen benchmark. So ideally with this, there's the ability to kind of expand the inclusion of what's in the database by accepting materials that have been screened through these other processes, to accept that data and present that data to the individuals that are using this as well. So that kind of that multiplies really the usefulness and the amount of reduces the amount of redundancy, I guess, when we can take equivalencies from other types of certifications as well. And then this is just a closer look at what happens when you click on one of those materials and you pull up, and you can see human environmental endpoints. You can see the range of them all there. This is the cradle to cradle view of it. And it also breaks it down by routes of exposure, oral, dermal, and inhalation. And then you can actually click on any of those to look at the toxicologist's rationale for providing that rating and what are the hazards associated with it. To just conclude, what's next? So again, as I mentioned, we are halfway through our collaborative. The pilot project is about halfway through as well. So really the next steps are kind of continuing both of those efforts within the collaborative, really continuing to educate stakeholders, to connect what initiatives are out there with what individual companies can do, and then really trying to develop that common agenda or a pathway or potentially a roadmap of how we can accelerate this, what next needs to be done. And then also with that, identifying, is there anything we can do as a group to provide resources or guidance, or is there anything that the industry can do to contribute to really having more clarity on what needs to be done in this space. And then with the pilot project, really the next steps are, after the alternatives have been verified and proved safer, then really demonstrating that, showing that to different organizations, showing its usefulness, and ideally that results in a widespread adoption so that more and more companies can use that to really pull that available chemical hazard data through and understand what is actually going into their different products. Really, the focus there is to accelerate that transition to safer alternatives. And so with that, I'd be happy to take any questions. I do have a couple of resources here, which are our websites as well as ChemForward's website. Stacey Glass at ChemForward, I'm sure would be happy to connect with any of you if you have further questions on their pilot project and what they're doing.
Great, Karen, thank you very much. I have one.
Sure.
So you've talked about finding ingredients that are safer. You use the word safer. And when I think of safer, I mean, this is a very general way, but I begin to think of how long is a piece of string, you know, because there are different levels of safety. So if you find an alternative that is safer, whatever that means, how do you then put it in a larger context? So for example, if you have a safer ingredient than is being used now, do you consider whether or not the less safe ingredient is safe enough? Do you consider the economic impacts of those changes? Do you consider whether going circular is the right thing to do? Because I've worked in consumer product areas where we've done life cycle analysis and found that putting something in a landfill is actually more environmentally friendly than trying to recycle it. So can you give us a sense for how you might assess the larger non-safety?
Absolutely. Well, I think that's a fantastic question. And I think the short answer is that no, I can't give you the answer right now, because I think that's something we need to figure out in this working group, in the industry, is really what level, like you said, that string, where are we putting the safer mark? And I think that's why we say safer instead of safe, because it's hard to verify. Yeah. And I don't have the background in toxicology to really understand where, in terms of, exposure and all of that, we can set that boundary. But I think that conversation really needs to happen. I think your point about circularity is really important to be part of the conversation too, because there are some things that may not contribute back to circularity in a safe way. And should it be landfilled? Should those things be landfilled during the time being, as we move away from those and use materials that don't have that same concern? There's a lot of different things that go into it, but I think that's a great point to really, and we've heard that from our members too, like what's the bar? Where are we setting this at as safer? And there's so much that goes into it, but yeah.
Could move to something safe, but there may not ultimately be a big picture benefit.
Yeah, there's so many trade-offs, absolutely.
Okay, so to take off on Leon's point, I think one has to be careful in terms of this moving to something safer, because sometimes the something safer means it appears safer because less is known about it.
Absolutely. I think that's a fantastic point. I think that is critical to consider. And yeah, I think I probably use that language a lot, moving to safer. There's a lot more to it, a lot more nuance, but I think that's definitely something that when we think about, what's the next PFAS, that's really what we're talking about. It is moving, is breaking that cycle where we can really move to things that are assessed and verified to have a high level of assurance that it's safe, but still understanding that we need to do the due diligence to look at everything that goes into it that maybe we're not seeing, that it's not on the restricted substance list yet, but why is that the case? Is there an issue? I think that's a fantastic question. I wish I had a better answer for you, but I do think that level of nuance is absolutely critical for this conversation.
Just real quickly, because what was safe yesterday, what was assessed yesterday using what the state of the art at that time is now being deemed not safe. Mm-hmm. And that material has gone through regulatory assessments. A lot of people have looked at some of those chemicals that are today, or people are questioning whether they're safe. And the question is, was it a problem with the chemical, or was it a problem with the approach we were using to do that assessment? So I don't, and it's not really a question, I guess, but that's something that we need to think about, because I think some of the problem chemicals you identified were at one point considered fine. Had lots of data generated on them, and they were considered safer than the product they were replacing. So
yeah, absolutely. It's such an important part of the conversation, and it just speaks to, I think, how big this challenge is to ensure that we don't continue to make those mistakes. But how do we do that with, there's a lot to it. And I wish I could speak more to the actual science behind it. Again, I lead this group, but I'm not the expert in it. But I think these are all really fantastic points that I know we'll continue to discuss in our group as well.
And I can maybe,
Kelly would probably have
make a quick comment regarding the standard behind the assessments, right? So I think there's enormous opportunity to advance the state of the science, because a lot of the existing paradigms are being applied. So factoring exposure, figuring out what went wrong with PFAS. You know, where did we fail there? Because we were using the state of the art at the time and even the state of the art that exists today. So I think there's a huge opportunity for everyone to engage in that.
Thanks, Kelly.
The same line, I think on comparative assessment, we should really have a bit of standard how we compare things consistently, and not just to be on the safe side. I mean, that's where we go to alternatives, accounting for what our colleague was mentioning, that some chemicals have less information than others. How do we handle that? I really like that we don't always take a conservative assumption, but we take best estimate plus uncertainty and then handle the two together. I think it's a way to go from.
Yeah, absolutely.
And where there's data gaps, a compound's not classified for that endpoint, but there's every effort made to fill the gaps with read across and all the tools we have available.
And that, like, how comprehensive is you have your database where you have the list of substance, you have the content, typical range of content in there?
So it is under development still. Is that what you're asking? How many materials are in there so far? Or
yeah, how many ingredients or chemicals, and would we have the percentage, typical percent contents for a certain function in there?
That's a great point. And I don't know. I would have to talk to my colleague at ChemForward because I don't actually manage the database. I'm just working with them for this stakeholder group. But I know that they're continuing to add things. And the ideal is that there's eventually a very high percentage of the materials that are in use so that there's the ability to look at everything and to make the most informed decisions. But it is in development still, so they're slowly adding to it.
We could find the students to run run our tools on such a database. All money if there is money, but students is elaborate.
Absolutely, yeah.
Possibility. I mean, so I have found recently that the organic composters who are handling that are sorting out these compostable packaging materials from their organic, and they're not being composted. And so the fact that they're being touted as this new great benefit to the environment, or they're just being landfilled. And when you landfill a degradable polymer, it's going to generate methane, and it's going to be probably worse for the environment. So I do think that we have lots of our clients who make that product. We've got to find a way to, and the reason that they just to complete it, the reason that the composters are not composting that material is they're trying to sell their organic compost, or their compost is organic. And if they have the plastic materials and the paper materials that have some plastic on them, or even just paper in their compost, they can't sell it organic, and that's a big gap. I think we have to solve that problem.
Absolutely, I think it's a fantastic point. I have a colleague, Olga Kachuk, who does a lot of fantastic research and convenes a different stakeholder group on compostable packaging, and it's really become apparent that there are certain uses for compostable packaging that make a lot of sense and contribute food product back into the compost system that the composters want. But then there's a lot of packaging that maybe doesn't make sense for that compostable to have that end of life and would be better served by being recyclable. But that's a big challenge. And then when you look at the composting infrastructure, it's kind of like these expanding bubbles of different challenges that need to be addressed. So when it's interesting to see the overlap of the different areas that we work on, but when you look at the whole picture of sustainability, there's so many different considerations, and that's a big one is composting and ensuring that the stuff that's designed to be composted does get composted, and the stuff that's designed to be recycled gets recycled. And there's a lot of gaps and dots that aren't connecting that need to be connected there for sure.
Other questions? It's been a really great discussion, so thank you very much.
Thank you. Thanks.
I'd just like to make a few closing comments, but before I do that, I wanted to begin by thanking all of our speakers today, and really for the informative presentations they gave. You know, I think we learned a lot today, and also I'd like to thank the audience for their attention and their participation, and we had really good engagement in terms of the speakers. You know, I feel like we just touched the tip of the iceberg around the complexities of packaging, but clearly I think we can all agree about the importance of packaging and the benefits, and especially as it relates to our food supply, because it's so important in terms of maintaining the safety and integrity of those products, and especially the way we market those today in terms of, you know, how we keep them on the shelves and making sure that they have extended shelf life. We also spent a good part of this morning talking about some of the advancements around packaging and some of the technological advances, especially coming down the pike on the horizon. Some of these are clearly capitalizing on the physical and chemical properties of nanomaterials, which I think are going to be very important in the future. Also, with those advancements, as we found out today, there's a lot of questions that come up in terms of, you know, human exposure and also the safety of some of these materials. Clearly, we had a number of presentations talking about migration of those materials into our food and beverages, and how we begin to model some of that so we can understand exposure. And again, you know, I really was very gratified to hear about all the discussions around exposure, because that ultimately, as you know, we talked in the last presentation and really throughout the day, you know, it's the exposure that really is what's important, because you know, from a toxicological standpoint, you know, everything can be toxic if you get to a certain dose. And Sam, you made the point that you know, most of these chemicals, you know, have a threshold. And so, you know, as long as we stay below that threshold, we should be safe. We also talked a little bit about the challenges surrounding new testing paradigms and approaches, especially in light of the fact that we're moving away from animal testing and some of the challenges around in vitro assays. Also, the regulatory landscape, especially when we think about it in a global perspective, is also very complicated, and there really is not the harmonization that we really need. And you know, if we think about many of the food companies, they are not selling products in one country; they are global, and the challenges that we face when regulations globally differ. I'm really happy we ended the symposium today talking about sustainability. You know, as I mentioned also in my opening remark, most of the stuff that I'm taking out to the curb every Tuesday morning, it's packaging materials. And we really need to figure out a way how to recycle some of these things, or at least reuse them in some way, and do it in an effective manner. And last, you know, I'm really happy that we once again had a presentation on communication. I think it was a very interesting strategy that was proposed, the 5 facts in 10 words, and how we might use that strategy to communicate our science. So with that, again, I'm really pleased we picked packaging as our theme for this year's meeting, and I think we all learned a lot from it. And again, thank you for your attention and participation. And again, thanks to all of our speakers. And everybody have a safe trip home.